Thank you. I just have a few slides I wanted to start with. Let me just let Lee get settled, and we'll get started.
Okay. All right. Thank you very much. Very great to meet you.
Yeah, nice to meet you. Okay. Thanks. Okay, we have two presenters for this session, and I just wanted to take a few moments, just as the chair prerogative, to just make a few comments about my perspective on the Herzenberg lab legacy in some ways. Let me just see how this goes. So I was a postdoc in the lab from 1994 to 2000, and had a great time. On the photos on this, of course, are highlighting the Kyoto Prize in 2008, and I always remember Len was always very adamant that it really was a prize to both Len and Lee. He always acknowledged that, so it was really a great honor for both of you. And you know, there really has been this huge legacy since 1959 when they first moved to Stanford. And it was really fun for me to look at the review of all of your publications. John, I think, put all of them online, and it's nice to see them all together, nearly 600 publications. And what I did in preparation for this meeting is just look through those, and the breadth is amazing. And I actually saw some of the very early publications and said, well, I would really love to read some of those again because they really look really nice. So, but it really is breadth and depth. And although, of course, one of their primary legacies is in B cells, but of course, me, and more as a T cell biologist, I saw even some of your early work was in T cells, suppressor T cells, and then, of course, there are publications on NK cells, all types of immune cell populations. So very nice to see that.
We did a true immunology lab.
Yes.
Equal parts.
Yes. Genetics and immunology, certainly both.
Although the T cells were always the dark side.
You considered it the dark side. I don't think Mario would consider it the dark side. Yes.
Wearing lipstick there.
And when I was looking through the LAH publications, I was reminded of this article. I just wanted everyone to be aware of this. This is really nicely written, Lee. And it was in 2013 in the Journal of Biological Chemistry. And I suggest if you haven't read it to go back and look at it. Really a really beautiful perspective about some of those early beginnings when you were working at the NIH and how you collaborated with some major folks in the immunology field. And of course, I focused on flow cytometry, and actually it's been an extreme benefit for me training with Len and Lee because flow cytometry has been the heart of all the research I've done since then. And it really has brought me into my current position, which I love at the Fred Hutch. And I'm really very proud of the fact that I really learned from the experts here at Stanford. The first publication I think most of us know about now in 1969 in Science. Fun to see that. And then, of course, there was the seminal publication in 1976 describing the technology to a more general audience. And what I love about this is that, you know, this is basically the same technology that's currently used. It's a very robust technology. It works so well that really most cell sorters, although we may hear about new technologies, David Voehringer is going to be talking about something a little bit later, but this really has been a mainstay in the flow cytometry field all these years. And then it's fun to see when we went to two-color flow cytometry. This was in 1977. And then three colors, bringing in a second laser. And that was also fun to see. That was in 1984. And then, of course, data analysis has always been a huge part of flow cytometry. And I should say that's one area where Lee, I think, always took a special interest in data analysis and working with, of course, Wayne Moore and David Parks, and this is one of this was in a chapter that was published, and just showing an example here of the contour plots and different ways of how do you display data, flow cytometric data. And again, some of this was really pioneered right here with this group. I also like to comment on the desk software, that software that we all used in the early days, and it was really ahead of its time. And the one aspect of it that it's a little bit funny for me to think about is that, you know, it had the automated way of standardizing the instruments. And it was interesting. I know when I left your lab and moved on to the NIH, at that time, the BD collection software did not have the standardization software yet there. So it came later, so we do have it now, but it was interesting there. And so for a long time, we were back from desk, which was automated, back to a manual system. So again, the standardization that we currently still use in our lab, actually. And then 8 colors, this is actually while I was in the lab, 8-color, 10-parameter flow cytometry. And then there's a publication the year later for 9 colors, including Nicole and, of course, Marty Bigos and many other people in the lab. And then I like to actually relatively quickly moving on from that, and I had left to work with Mario at the NIH, and continued to work with Mario to expand the capability of flow cytometry. And where we currently are with some of the technology that actually Mario worked with BD on to prototype, to be able to allow us to look at 28 markers simultaneously. And my lab has actually now published several of these panels in cytometry that really work beautifully. Again, really demonstrating the robust qualities of flow cytometry. And then, again, I want to highlight data, again, analysis, and data display this logical, or what people sometimes refer to as bi-exponential transformations. And, I put that bullet off to the right. When I first learned of this, and I really think it's true, I think this is really one of the most impactful innovations in looking at flow data. I mean, it's amazing. I mean, I can't think of looking at flow data without this. So this was really a dramatic change in the flow cytometry field, in my opinion.
It's a language, and
Yeah.
comparable to language.
Yeah, I think you're right. It's just a dramatic, it's just that, a shift, a paradigm shift in some ways, but really a beautiful.
If you think that was impactful, go back to the day when we looked at everything in linear.
Okay, yes, wow, okay.
We did not have log.
Yes, that's a good perspective to have.
It was amazing. There were the people who rejected logic, and the other people who jumped for it.
Wow, that's funny. I had not thought about that in the past. And then this very nice publication that I think Lee had led along with David Parks about this interpreting flow cytometry data. And again, I think this is really impactful for you guys to really help inform how do you look at flow cytometry data and what do you look for. And related to that, a colleague of mine at the Fred Hutch actually published a follow-up on that, or what he called an update in 2021. Florian Mayer, who at the time he was a postdoc working with Martin Prilich, and then Thomas Lechte at the NIH working closely with Mario. And I think it was really very, it was an honor to you, Lee, that they actually named their article an update on the guide, because they really do, I think, recognize that you guys had really set the stage for thinking about informing the general scientific community about how to interpret flow cytometry data. So that's why I just wanted to highlight that. And I think this is the last publication, this was that I'm highlighting here, and this was from Daria when she was in the lab, and relatively recently, and I think Wayne is going to actually follow up on a little more about this, but Lee continues to pioneer new ways of looking at flow data, and it's exciting that this is continuing. And I think that was it. So thank you very much. I think we can put up the slides then for our next presenter, who is Hiro. And thank you, Hiro, for being here, and we look forward to hearing your talk.
Okay, so good afternoon. So my name is Hiro Nakauchi. I may not, some of you may not know me, so just briefly, I was a postdoc at Stanford University in Len and Lee's lab long time ago, 1983 to 1985. And then following year, I came back as a visiting scholar for the summer. So at that time, on the left, you can see Len and my son, Yusuke and Taka. That was 1985. And then 2010, they're, 30s, I guess, early 30s. Again, with Len and Taka, Yusuke. And now they're living all nearby. And Yusuke is a hematology oncologist, a lecturer at Stanford University. And he works in a lab on the same floor, same building. And also Taka's family is also living in nearby in Los Altos Hills. So we all, Nakauchi family, live very close. So that's one of the best part of my life here. So when I was a postdoc, what I did is I isolated CD8 alpha chain gene. At that time, it was called LYT2. And following this, I learned molecular biology here, so I cloned many other genes later. What I learned here, many things, of course, but actually I was one of the users of first fax machine in Japan under the guidance of Professor Ko Okumura, who was a former postdoc in Len and Lee's lab, as some of you may know. And so I was kind of expert in Japan at that time, but when I came here, I was very shocked by the, not just the machine, but also the setup and data analysis, and also the people, supporting all these facilities. So I was very surprised and impressed by the fax, facility here. But scientifically, the moment I truly became interested in fax machine is when I observed David one Saturday morning, I guess, before or after I went to a tennis court. I thought, he was trying to set up an automated cell deposition unit at that time. It was called ACDU. And it was such a marvelous idea, I thought. So I started to think about using this system in my science afterward. So that was really impactful experience for me. So after going back to Japan, I started my own lab, and what I did is single-cell biology. And my target was from T lymphocytes. I changed from T lymphocytes to hematopoietic stem cells. The reason is, I would say, because rag knockout mice can survive. People can survive without T cells and B cells, but people cannot survive without hematopoietic stem cells. So I thought, HSCs are more fundamental in this, maybe a hematopoietic system. So at that time, my interest was to see if HSCs really self-renew or not. Al Weissman and others said that HSC can self-renew. But if you think, here's a HSC, and if you think in a clonal level, what happens if they only do asymmetric self-linear division or symmetric self-renewal division or differentiation? So if you think, if you transplant a single HSC, what could happen? If this is the case, no mature hematopoietic cells, right? And if this is the case, we lose, hematopoietic cells and HSCs. And if this is the case, asymmetric cell division, it's really nice because we can, always supply blood cells, and also we can maintain HSC. And interestingly, with this system, we should be able to make the number of HSC constant. It's very nice, right? Nice system. But to prove this, I thought we need to do a single-cell transplantation of hematopoietic stem cells. Now, that's what I did a long time ago. Still the most cited paper in my, the papers I have published so far. So this is the results of after the single-cell HSC transplantation. Essentially, all myeloid and lymphoid cells were reconstituted by single HSC transplantation. And interestingly, unlike human HSCs, they were CD34 negative. So this took extra 6 months or so to publish because people didn't believe it. But anyway, so this was the results published in Science some years ago. So this means that there must be some kind of self-renewal division. Still not very clear, but first evidence, experimental proof that HSCs can maybe self-renew and differentiate in different lineages. So, however, when we did this and we published this paper, I was more or less confident that we should be able to expand HSCs in culture, ex vivo expansion. But it was very difficult to do that. We tried all available lymphokines, cytokines, but we couldn't expand. So I just doubt whether HSCs really self-renew. Or it may be that we have many HSCs in the beginning. They use weekly or daily one of them to maintain our hematopoietic system. So I did a very simple, practically difficult, but simple experiment. That is, we transplanted single HSCs and waited for about 3 months to reconstitute the entire hematopoietic system. And then after that, we recovered all the bone marrow cells, many bone marrow cells, and transplanted in secondary recipients and performed limiting dilution assay. So in this experiment, only the first single cell, we used immunophenotype by fax and sorting. But after that, this portion, nothing to do with the phenotype, just a functional assay. And then we found that we started from 1. CRU means number of functional HSCs. So start from 1, it's very easy to count. So 3 months later, when we measure the number of HSCs, so as you can see, many HSCs, functional HSCs, so there's very clear evidence that HSCs can self-renew in the bone marrow in vivo. LNK knockout mice, by the way, LNK is a negative regulator of self-renewal signal. So they have much larger number of HSCs, as you can see here. But anyway, in any case, HSCs can expand, self-renew extensively in vivo. So we and many others turned our attention to bone marrow niche because we believe that some special mechanism in the bone marrow niche so that HSCs can self-renew, right? We cannot do it ex vivo, but they expand in vivo. So many people started working in bone marrow niche. So over the years, I learned, we did many HSC transplantation. I learned that in order to engraft donor HSCs, you must empty the bone marrow niche. Without emptying HSC niche, there's no donor HSC engraftment. So we need a space for donor HSCs to land and survive. So this is something I learned. Now I turn to the project to hematopoietic system to organ generation. So some of you know Jianzu Chen. He is now a professor at MIT. He published after graduating Stanford from Herzenberg lab. He moved to MIT or Harvard, I think, to Fred Art's lab and published this very interesting paper, at least to me, that is LAG2 deficient blast deficient blood cell complementation, an assay for gene function and in lymphocyte development. I found this paper very interesting, as I said, but from a different viewpoint. So let me explain what he published. So he used RAG2 knockout mouse. The RAG2 knockout, as you all know, there's no T cells, no B cells, because they cannot rearrange immunoglobulin T cell receptor genes. So what he did is he obtained embryo from RAG2 knockout mouse and injected wild type ES cells to this embryo to make a chimera. So he's got chimera, and he's got both T cells and B cells from the donor ES cells. So then he tried to use JH, as you know, heavy chain joining region, heavy chain J region deficient HSCs. Then what he found was quite interesting. He was able to see generation of T cells, but not B cells. So this clearly, more or less, indicates that JH gene segment is important for the development of B cells, but not T cells. So he used this blood cell complementation to assess the function of a gene in lymphocyte development. A very nice idea, I think. But I thought, I took this from a different angle as a stem cell biologist. I thought this in this RAG2 knockout embryos, niche space for T and B cells is wide open, so that donor ES cell-derived cells can use this niche space and expand to compensate the defect in this RAG2 knockout embryos. So this is how I got the idea of this organ niche and organogenesis using this organ niche. So if there's an empty organ niche, for example, in kidneys, then if we make chimeras, we can generate kidneys, mainly, actually, all of these cells making these kidneys should come from injected donor ES cells, because they are the only cells who can differentiate into kidney cells, but the host cannot. So I had this idea many years, and finally I found some good student who actually tried this experiment. So this is the SAL1 knockout mouse. SAL1 is an important transcription factor for the development of kidneys. So these knockout mice cannot form kidneys. But when Toshi Kobayashi injected GFP-marked ES cells into the embryo of this SAL1 knockout mouse, then we were able to observe, as expected, as we hoped, the generation of kidneys here. And as you can see, these kidneys are very, very bright for GFP, indicating that these kidneys are made of cells derived from ES cells. So this is a way we can make stem cell-derived organs. So this was nice, but our future goal, of course, is to make human organs. So we cannot make organ-deficient human embryos. So instead, we are thinking of using livestock animals like pigs or sheep. So the idea is, if here's a patient with end-stage, say, heart failure, then we generate iPS cells from this patient, and then inject these iPS cells into the blood cyst of organogenesis disabled animals, like heart-deficient embryos. And then, if it works, we call it interspecies organ complementation. The previous one is intraspecies, mouse to mouse, but now this is the interspecies complementation. So if it works, we should see a birth of a chimeric pig that has a heart, and this heart should all derived from injected patient's own iPS cells. So we can, if it works, we can take this heart out and transplant back to this patient, because the heart, although it was formed in pig body, the heart cells are from patient's own iPS cells. So it should be a autologous transplantation, and we don't need to worry about rejection and so on. So next we try to see if blast complementation works across species, interspecies blast complementation. So we used rat and mouse. They're both rodents, but they are different species. They cannot mate. They have different chromosome numbers. And rat, as you can see, 10 times bigger than mouse. So we try to see if we can generate mouse organs in rat or rat organs in mouse. So we did that experiment first. This was like a 2010 paper, a long time ago now. We used, for this experiment, PDX1 knockout mouse embryos, and then we injected wild-type rat ES cells, or iPS cells, and then we were able to see chimeras. That was good. And these chimeras have rat pancreas. But interestingly, these interspecies rat to mouse chimeras were somehow mouse-sized, and so was the rat pancreas. So although pancreas was made of rat cells, the size was mouse-sized. Very interesting. So it's too small to transplant back to rat. So we performed the opposite experiment. That is, we generated PDX1 knockout rat, apancreatic rats, and we injected wild-type mouse iPS cells into this embryo. And as expected, we were able to generate chimeras, but this time the chimera was rat-sized. And pancreas was made of mouse cells, but again, the pancreas was rat-sized. So it's huge pancreas, as you can see here. So this is the mouse pancreas generated in rats, and this is a wild-type control mouse pancreas. So this is apparently too big to transplant to mouse. So instead, we performed islet transplantation, like we do for human diabetic patients. And we isolated islets from this gigantic mouse pancreas and transplanted only 100 islets to the drug-induced diabetic mice with the hope that these islets can cure diabetes without any immunological rejection. So that is exactly what we found. Within 3 months, these diabetic mice, their blood glucose levels returned to normal levels for over 300 days. And within 3 months, their glucose tolerance test returned to normal. And after 6 months, there was no remaining rat cells in the graft because recipient mice are immunocompetent, obviously, and they're able to clearly discriminate rat cells, contaminating rat cells from their own mouse cells. So, and when we removed these grafts, these blood glucose levels went up very high, indicating that these transplanted 100 islets, only 100 islets, could actually essentially cure the diabetes. So importantly, no long-term immunosuppression necessary. Obviously, these are autologous transplantation. So this is the proof of concept data for our ultimate goal, interspecies organ generation. So we also generated apancreatic pig embryos, and we performed essentially the same experiment, and we were able to complement this defect. And these should be a lethal phenotype, but they survived with the exogenous pancreas. This is pig to pig, not human yet. So this is nice. We showed that we're able to do the same, apply the same principle to large animals like pigs. However, at this point, I should mention, Len kindly communicated this paper to PNAS. That was good. But at this point, we were able to generate human ES cells or iPS cells to these apancreatic pig embryos to see if we can generate human pancreas or not. But unfortunately, at that time in Japan, implantation of human animal admix embryos to in vivo was not allowed. So essentially, you couldn't do this experiment in Japan. So luckily, I had an offer from Stanford University, so I decided to move from University of Tokyo to Stanford University. And I appreciate Len, and they, of course, supported my transition to Stanford. So anyway, so far, we have generated many different organs as shown here, allogenic and xenogenic, and also allogenic in pigs. And in pigs, we have even tried to generate both pancreas and blood vessels in this double knockout pig embryos. So this works nicely. So now we have sufficient data to do human-animal chimera studies. So in this country, it took some time to get permission, but now we're able to do this kind of experiment. We, in collaboration with scientists at UC Davis, we made human sheep embryo chimeras and injected into the uterus of the foster sheep mother. As you can see here, this is the uterus, and making a hole, and we're injecting these human sheep chimeric embryos. And 3 weeks later, we have recovered all these embryos, and using digital droplet PCR, we quantitatively measured the amount of human DNA, and we estimated that only 1 out of 8,000 cells are human. So clearly, it didn't work like mouse and rats. So this is the difficulty, and we have made a lot of progress. But briefly, we have prepared organ niche by brushless complementation. We have also another approach that is cell-competitive niche using IGF-1 receptor knockout host. Also, we can do in utero transplantation. But unfortunately, for human mouse chimeras, there's a barrier earlier than this between E6 and E10. The organogenesis starts about E10. So this barrier is even before the ideas that we developed. So now we're working on this issue, how we can get over this xeno barrier by modifying human iPS cells and also host as well. So this is the current status of this human-animal chimera work, and hopefully we'll be able to soon generate human organs in pigs or sheep or whatever. So this is what we are in terms of basic research. We're trying to generate organs from iPS cells. We consider this as a next generation regenerative medicine. And then we have a little more translational projects already in progress. For example, I mentioned that it was difficult to expand hematopoietic stem cells ex vivo. But somehow we finally, after more than 25 years of research, we found a way to expand mouse hematopoietic stem cell first, published in 4 years ago. And now recently, just this year, we published the method to expand human HSCs ex vivo. So this is very new. And technology transferred to a company, and this company is trying to do clinical trial. Also, a little more advanced to the translation, I thought about, we have developed a system to generate antigen-specific T cells from iPS cells, patients' own iPS cells. So this is perfect because, as you know, T cells require MHC recognition, MHC match. But with the iPS cells, if we can generate T cells, there's no issue about that. So that was my idea. And we published this first almost 10 years ago. now this technology has been transferred to a company, Century Therapeutics. This is a US company, and they are trying to do clinical trials. And these are the people used to be in my lab, and now they are independent and continue their work on, in this case, hematopoietic stem cells and also T-cell therapy. And most advanced is the platelets from iPS cells. The blood transfusion is a very important clinical interventions. But still, we are totally dependent on donation of blood. And platelets, we cannot store these cells, platelets frozen. So it has to be fresh, always fresh blood from donation. So, but if we can generate platelets from stem cells like iPS cells or ES cells, we don't need to worry about the supply of platelets. So that was the idea. First publication was like 2008. And this technology was transferred to a company called Megakaryon, 2011. And now it took a long time, but they started clinical trials in Japan to 2022. So, it takes really time to translate these discoveries or technologies of basic science, but, I'm quite happy with this progress. So now I change my topic again. So as people have already shown, I think the fax is a great invention by Len and Lee, and this is a photo with Len, Lee, and also Tasuku Honjo, more recent Nobel Prize winner in Kyoto. So I had a long experience of using as a user of the fax machine, but having long been troubled by the unreliability, non-stability of fax, particularly sorting, so I wanted to develop a sorter that'll be trouble-free and easy to use. And particularly, I thought, Japan has all the technologies, electronics, computer science, lasers, what else, lenses, and so on. So I was quite convinced that Japan should make a good, reliable, easy to use fax like home appliances. So I asked Sony, through my high school classmate, to develop a fax that anyone can use and that'll be trouble-free, very easy to use. Luckily, we're able to do this. So some Sony people, for example, is here, visited Stanford, and also Len and Lee visited Sony headquarters in Shinagawa, Japan, some time ago. So obviously Sony has technologies to do this, and within 3-4 years, I think they had a prototype, and it took some time after that to commercialize it, but finally they were able to commercialize their products. So this is Ko Okumura, and Lee and Len, and this is myself, and Len gave a seminar at Sony headquarters. So this was at the Atsugi Institute, Sony's institute, and they had already a prototype at this point. This is my high school friend. This is my fax for manager. So we visited and we exchanged ideas and so on. So it took some time, but now they were able to commercialize the machine, and they are growing very rapidly, obviously, very easy to use, trouble-free, and so I'm quite happy in this development of fax in Japan. So now the most recent one, as far as I know, is this GMP compatible cell sorter. So I think this is, I've never used it, but I think it's okay, very reliable. Use it, good. Yeah, it's always drops, at the same point. That makes a difference from other sorters. Oops. So in our institute at Stem Cell, we also have one, and we're using it. So I'm quite happy with this development. So finally, I think Herzenberg lab is a niche for young scientists. Used to be. And I learned many things, but that was the best time in my life as a scientist particularly, but also, my family is a part of Herzenberg family. All my sons, grandkids, they're happy. So I think we should, or I should, keep this tradition, legacy, and to the next generation. Yes. So thank you, Lee and Len. Any questions? Yes.
So Hiro, may I? Yeah. Great talk as usual, and very enjoyable reading for the whole story. Now, on the xeno barrier, of course, you've done a huge amount of work on that. It's very easy to think about MHC complementation. That's fine. But it looks to me like, as you certainly thought, it could be much more complex. You can think about receptors for hormones that may be different. You can think about adhesion molecules. You can think about, and you've done a huge amount of work on making it compatible, subtle differences in development. As you know, for example, human genetic diseases not always are recapitulated by mouse knockouts. And what is your thought about that? I noticed that the list of things you want to do at the end, but give us an idea.
Yeah, I totally agree with you. Are you suggesting that I should give up this project and do something else?
No, you did too well in the past. So please keep going.
Well, I'd say 15 years ago, people never thought that we can make rat pancreas in mouse, right?
I know.
So there must be many barriers, many difficulties we have to overcome. But somehow I learned, biology or organism is quite flexible sometimes. And we're just giving, making progress step by step. The only problem in this case is like hematopoietic stem cell study before we were able to expand. We only have small number of ear cells to be injected in the process. So we cannot do a whole, genome CRISPR screening, or we cannot do large-scale screening for this. So that is the difficulty, but it's also a challenge. It's fun, so I enjoy that.
Yeah, really beautiful talk. Thank you for taking us through that. You make it sound so easy. You just put an iPSC cell in an animal and it's going to grow the organ that you want. What do you see as some of the biggest challenges or pitfalls of some of these methods? Like, do the organs completely work the way you want them to? Are there some gaps that are kind of concerning you?
Yeah, initially I thought it would be not that difficult, because if you look at embryos, human embryos and pig embryos, mouse embryos, they all look similar, right? At least in the first 6 days or 10 days. But actually, that time period is very critical for outlier cells. So they have a very unique system to recognize and eliminate those cells. Now we're working on it, now we're getting some idea. So clearly it's before macrophages or NK cells. It's before, the hematopoietic system develops. So there must be some pretty innate immune-like cells there, or there must be some still unknown mechanism to recognize, eliminate, or sort out to the placenta of the outlier cells. So that we're really enjoying working on this. So, it may take another 25 years, like hematopoietic stem cells, but we are learning a lot in terms of biology. So it may take some time to generate human organs, but we can still contribute to biology, I think. Yeah. Yes?
I have a question about your xenobarrier. So is it an immunological problem or a developmental problem?
Yeah, I think.
Do we know that?
Clearly, what I can say is it's not a conventional acquired immunity, because way before that, way before those hematopoietic cells develop. So it has to be some kind of innate immunity, very primitive macrophages, or some myeloid cells. And also it could be a developmental issue. As Saverio mentioned, the affinity of cytokine receptors could be different due to the evolutionary distance between human and animals.
Yes.
So many factors could be cell cycle issue, proliferation issue, but eventually we're getting better though. So we are making human mouse chimeras in certain areas, not systemic. So I cannot answer all of them, but eventually we'll learn, what is this xenobarrier.
Yeah, the reason I asked this question is this: if it is immunological problem, you can use a long-term immunosuppression to the recipient, the blastocyst recipient. Probably you can circumvent that.
Yeah, we have already used NSG mice,
Okay.
islet receptor gamma knockouts. As I said, it's before the immune system develops. And according to the textbook, because those rat cells in a mouse embryo are there when at the time of immune system is developed. So they should be tolerant to those mouse, but it doesn't work that way.
Okay.
Very interesting.
Yeah, thank you.
Thanks again, Hiro. It was a great talk, very fascinating. And our next speaker, sorry about that, is Yang, and we look forward to hearing your talk, Yang.
I need to look at that. Good afternoon, everyone. So I'm sorry, I didn't prepare emotionally for some slides, but just I think I may focus on the science progress in the lab. So I will talk about the most recent surprising finding because the lab is studying the B1 cells. So it's the earlier developing primitive B cells in the mouse. So we find that they play a surprising antigen-specific role in the induction and maintenance of the immune tolerance. Importantly, this function is not mediated by their antibody they produced, but rather mediated by the antigen-specific antigen-presenting function. So the study from the past decades have well established that the mouse B cell development occur in waves and in the different anatomic site. So the earliest the B cell progenitor can be traced back to the embryo day 9 in the yolk sac and the AGM. And this progenitor is prior to the emergence of the hematopoietic stem cell. And they give rise to the earliest wave of the B cells called the B1 cells. And after birth, the B1 development largely diminish, and the B cell development is dominated by the conventional B cell generation from the hematopoietic stem cells. So after the short period of the de novo generation of the B1 cells, they undergo antigenic selection and self-replenishment. So finally, comprising a small but functional important subset in adult throughout of the life. So their expression, their developmental pattern is akin to that of the chicken B cells. Because of that, it's called the primitive B cells found in the mouse. In the field, also people like to view them as innate-like B cells. So the finding that the B1 and the B2 arise from the distinct progenitor appear at a different timing during ontogeny, led Lee and Len back in 80s proposed the concept of the layered immune system, so in which the B1 and B2 evolve sequentially to give the progressively more functional advanced immune B cells. Actually, to be right at that time, this concept is really controversial, but now and more and more accepted by the field. And this concept is expanded to the other immune cells like the macrophage and the T cells. So it's really having a vision at that earlier time. It's really, so glad I can see the latest publication really emphasize the idea of the layered immune system. So compared to the conventional B cells, B1 have the characteristic presentation in the lymph node organ, so that they are very real in the lymph node, but constituted as a very small subset in the spleen. They are the dominant B cell in the body cavity, like the peritoneal cavity and the pleural cavity. And also, the study by France back to the 80s, and also recently, they're also showing the substantial proportion of the IgA-producing B cells actually is derived from, in the gut, derived from the B1 cells. And also, intrinsically, thymus also contain the B cells, and many of them expressing CD5. Now we find that these CD5 positive B cells in the thymus actually are B1 cells, and they play a very important role in the induction of the immune tolerance. That's one of the study I will talk about today. So in addition to the producing antibody, so our study showing that these earlier developing primitive B cells play a key role in the induction and the maintenance of the immune tolerance as antigen-specific antigen-presenting cells. So I will talk about, but importantly, their antigen-presenting capability is determined by when and where they reside. So in the neonatal thymus, we found that the B1A undergo robust activation and differentiation to the air-expressing antigen-presenting cells that play a key role in the neonatal tolerance induction. So this is an unpublished study. So because of that, we just got an R01 grant, which can support us for additional couple of years study in the Herzenberg lab. And also, on the other hand, recently we also published a paper showing that in the adult spleen, the CTO4, which is a very key T cell inhibitor, the CTO4 expression in the B1 constrain B1A differentiation to the antigen-presenting cells. Without this expression, without this control, the immune homeostasis actually is impaired. So the B cells function is largely mediated by their BCR. So here just showing the organization of the Ig heaven chain VDJ complement constant gene segment in the mouse chromosome. So their mouse have a very large VH gene, so they're about 100 genes belonging to 15 families. So the most distal family gene is called VH1. This is the largest family, which contain half of the VH gene VH genes. So these are the genes are dominantly expressed by the B2 cells. And a couple of years ago, we conduct a large-scale quantitative IgH sequence comparing the B1 and the B2 subset. Actually, we find that both B1A and the B2 preferentially using the VH1 families, although there's a certain VH genes are statistically expressed on the B1 versus B2, but yeah. And so the primary Ig heaven chain diversity is generated during the B cell development. So first step is the RAG-mediated VDJ random rearrangement. The IgH diversity can be further augmented by the nucleotide deletion and addition in the DJ and the VDJ junction. Also including the TDT enzyme-mediated nucleotide addition. So the Ig heaven chain CDR3 region contain all of the diversity information generated during this process. So because of that, the CDR3 play a very important role in defining the antibody affinity and the specificity. So the potential number of the different antigen binding sites is estimated to be enormous. So if each cell have an equal chance to using the VDJ recombination, the B cells have an identical CDR3 should be in the one mouse or across the mouse should be very, very few. But surprisingly, so that's not the case for the B1 repertoires. So this is showing the Ig heaven chain CDR3 nucleotide tree maps. We comparing the B1 repertoire CDR3 with the other B cell subset like the B2 and the marginal B cells. So as you can, so each plot, each spot in the plot indicate the unique CDR3 nucleotide sequence. So the size of the spot refer to the relative frequency of that sequence. So as you can appreciate that compared to the B2 and the marginal B cells, the B1 repertoire, no matter it's in the spleen or the peritoneal cavity, they are less diverse and more repetitive. So this just showing, we quantify the CDR3 diversity, so you can, this is showing they are statistically less diverse compared to the B2 and the marginal B cell subset. And we also track the B1 repertoire from their neonatal period to their long-term residency in the adult animals. So we can show that their repertoire undergo potent selection during the ontogeny. So the recurring sequence, so like the spot, they didn't appear until mice reach adulthood, and their frequency increases as the mice get old. So this is a typical prototype. Is the VH11. It's the PTC binding clone. You can see that trend. And then the selection is not only occur in the individual mice. We also can identify a ring of the identical VDJ sequences which encode an identical peptide. They can positively select it in the adult B1 repertoire and shared across the mice. So that this sequence we reason that they represent evolutionary selected conserved structure selected into the B1 repertoire. So consistent with their unique repertoire, the B1, one thing we forgot to say that. So we also find in the repertoire selection proceed in the germ-free mice. So the ruling out, selection is not mediated by the microbiota-derived antigen. Since the B1 antibody is polyreactive and tend to bind the self-antigen, so like the carbohydrate, glycolipid, and the glycoprotein. So that suggests that this selection is likely mediated by the self-antigen. And then the evidence showing the B1 is repertoire is positively selected by the self-antigen is evident by the study by Kyoko and Randy. So they published a very elegant study showing that the B1A with the IG specific for Cy1 glycoprotein are positively selected by the Cy1 antigens. So, consistent with their repertoire, unique repertoire, the B1 are well known to produce natural autoreactive antibody. So this antibody play a very important role in both immune defense and the homeostasis function. So they clear the cellular metabolic debris, and they also provide the barrier for the organ transplantation. And they also participate in the front line of the immune defense to the infections. So this also raises the question regarding how their autoreactive antibody is regulated, because there should be the regulation to prevent the BCR-mediated self-antigen presentation and the high-affinity autoantibody producing germinal center formation. So we find one of the key of the controller is the CTLA-4 molecule. So CTLA-4 is the key T-cell co-inhibitor. It's also the first antibody-targeted T-cell checkpoint blockade in the cancer immunotherapy. So years ago, we surprisingly find actually CTLA-4 is selectively expressed B1 within the resting B-cell compartment. So here just showing the CTLA-4 protein expression by FACS, we compare the B1 with the other B-cell subset. So you can clearly showing that the B1 expression low level in the CTLA-4, but other B2 do not express. So this expression is not mediated by the transfer from the CTLA-4 to the T cell, from T cells to B1, because they also showing in the T cell deficient mice. And consistent with the study showing the human BCL-AL also expression CTLA-4. So we got the EU TCL transgenic mice from Tom Keep's lab. So we're showing that they also, this is a mouse model for the human BCL-AL, so in which the BCL-AL-like tumor are derived from the B1 cells. So we're showing that actually CTLA-4 expression is increased in the B1-derived tumor, but not do not express in the B2 in the same mice. So we try to understand what's the function of the CTLA-4 expression on the B1 cells. So we generate the B cells conditional knockout. So the CTLA-4 is selectively deleted in the B cells, but remain intact in the T cells. So we find that the IgM high B1 cells from the CTLA-4 conditional knockout mice expression increases in the BCR signaling, and they proliferate, and compared to the CTLA-4 expression counterpart. But in the meantime, this activated B1 cells further internalize IgM. So, and these IgM negative B1 cells in the CTLA-4 conditional knockout mice, they downregulate the BCR signaling, they stop the proliferation, but instead they upregulate the genes program for the antigen-presenting cells program, like the transport, MHC expression, and the protein folding, and the other program characteristic of an antigen-presenting cell gene signature. So, and this activated B1 derived the antigen-presenting cells. If you transfer these cells into the normal mice, they can induce T follicular helper cells and a germinal center response in the normal, in the spleen of the normal recipient. So, yeah, I don't go to the detail, just showing the conclusion. And consistently in the B-cell CTLA-4 conditional knockout mice, they generate autoreactive antibody, which including the antibody reacting to the array of the carbohydrate autoantigens And the mice also develop the autoimmune syndrome when the mice get older. So this study suggests that the CTLA4 expression in the B1 constrain their activation to become the antigen-specific antigen-presenting cells. And this control is very essential to maintain the immune tolerance. And so in contrast to the regular B1 in the spleen and the peritoneal cavity, so their antigen-presenting cell capability is prevented. We surprisingly, the recent study, we surprisingly find actually B1 emerge as the earlier wave of the tolerance-inducing antigen-presenting cells in the neonatal thymus. So thymus is the central lymph node organ. So the developing T cells have to go through the selection process before they migrate out to the peripheral to become the mature T cells. So what we find is that in the neonatal thymus, B1A induce air expression and differentiate to the antigen-presenting cells. And they promote the generation of the B cell tolerant T regulatory T cells. And these T regulatory T cells suppress the B cell responsive CD4 T cells, thereby enabling the establishment of the B cells in the peripheral. So this is the finding currently support this model. We haven't published this data yet, but it's very exciting. And so the goal of the TCR selection is that they will select the TCR which can recognize the non-self but tolerant to the own cells. So this is largely mediated by the recognition of the self-MHC and the peptide presentation by the thymic antigen-presenting cells. So the fate of the T cell selection is really dependent on determined by their affinity model. So majority of the T cell will subject to death because they express useless TCR, cannot recognize the self-MHC molecule. Only a small portion of T cells they are further selected. So if they express in a weak affinity, they will select either into CD4 or the CD8. But their TCR is relatively high affinity. They will either delete these autoreactive T cells or redirect these autoreactive T cells to the Treg lineage. So the really milestone, the breakthrough in the central tolerance is the finding that air promote the tissue-restricted self-antigen expression and the presentation in the thymic antigen-presenting cells. So the most characterized antigen-presenting cells in the thymus is the dendritic cells and also medulla epithelial cells. So as the air is the transcription factor, so these antigen-presenting cells induce air. So air can induce the tissue-restricted self-antigen in these cells. In this way, they can present this antigen to the autoreactive T cells to either delete the T cells or divert to the Treg generations. So very interesting that the air can induce a large number of the tissue-restricted antigen expression. So it has been shown that each cell the air can induce a subset of the TRA per cells. And also B cells actually, it's a classic, can be a potent antigen-presenting cells. If their antigen presentation is mediated by the BCR, their efficiency is much, much higher than the classic antigen-presenting cells like the macrophage and the dendritic cells. So, and also the air defect in the human or deficient in the mouse, they all have the multi-organ severe autoimmunity. We just analyzed the neonatal thymus. We surprisingly find CD5 positive B cells, which including the IgM high and IgM negative, IgD negative population. They are readily detectable in the day zero in the newborn thymus, but they increase markedly after birth in the first 1 week. And surprisingly, this CD5 wave does not occur in the TCR knockout mice. And so the rapid rise of the CD5 positive B cells in the neonatal thymus is because they undergo robust proliferation and differentiation. So they proliferate. They also auto-induce air expression, and they differentiate to the activated cells, the IgM negative CD95 and the CD80 high and MHC high, which is in the phenotype, it's the same phenotype as the IgM negative, IgD negative B1 in the CTLA4 deficient spleen, antigen presenting cells. And we also analyze a bunch of the knockout and TCR transgenic mice, or using the antibody to block the certain signaling. So we find that this activation requires CD4 T cells. Not only the CD4 T cells, they also require the T cells expression the appropriate TCR, because if you're using the CD4 T cells transgenic mice, it's still you can't see the activation. So suggests that they somehow require very certain CD4 T cells expressing certain repertoire. And also they depend on the CD4 T cells mediated CD40 signalings. So this suggests that their activation is triggered by their specific interacting with the CD4 T cells in the neonatal thymus. So here just showing the air induction. So we using the air GFP reporter mice. So as you can see here, the air is induced at a high level in the CD5, both IgM high and IgM negative CD5 positive cells in the neonatal thymus. But and then the air expression and actually correlated with the activation, because the expression level is higher in the IgM negative, IgD negative, CD5 positive subset, and also correlated with the CD95 expression. And but the expression is downregulated as the mice get reach the adulthood. I will talk about later. And so we also using the cell transfer to trace to identity the origin of the CD5 B cells, because it's also long-term argument. Are these CD5 positive B cells in the thymus are really B1 cells, or they just the B cells upregulate CD5 on the surface? So we transfer bone marrow progenitor and also self-replenish B1 cells. This can using the different anotype marker, also congenic marker. So in this way, we can distinguish in the donor. So we transfer to the day 1 B cell deficient MUMT mice. So after the 2 months, we analyze when the mice get B cell marrow. So we analyze the spleen and the PERK thymus. So we analyze the thymus. We can get so you can see that the B cell will reconstitute in the thymus of the MUMT mice. But if you get on the peritoneal B1 donor, you can clearly showing that majority of the CD5, no matter it's an IgM high or IgM negative, are largely derived from the transferred B1, but minimally derived from the bone marrow progenitors. So that indicate the CD5 B cells in the thymus, they are B1 cells. They are generated independent from the bone marrow B cell development. And so we're also showing that the IgM negative, IgD negative air expression CD5 B cells are the progeny of the activated IgM high B1 cells. So we're using a similar strategy. So we transfer the B1. I forgot to mention that we only detect the B1 expression air in the thymus. In the B1 in the peritoneal cavity and the spleen, they do not expression air. So we transfer B1 from the peritoneal cavity, which is the air negative cells from the air reporter mice, together with the bone marrow, we transfer into the day 1 MUMT mice. Then we analyze the 19 days after transfer, because we know air expression in the adult thymus will downregulate. So we will try to analyze the earlier time. So you can see that we get the B cell reconstituted in the MUMT mice. We track the peritoneal B1 derived donor. So you can see that they give rise to the IgM negative, IgD negative population. And importantly, this population expression error. So it's mimic what's happening in the normal mice thymus. So really indicate the thymic IgM negative, IgD negative air expression CD5 positive B cells are derived from the activated IgM high B1 cells. So yeah, so that means the air negative IgM high peritoneal B1 they gave rise to the IgM negative IgD, IgM negative IgD negative air expression B cells in the recipient thymus. So importantly, this activation only occurs during the neonatal window. So this including the proliferation. So we're doing the in vivo BRDU cooperation. So you can clearly see that substantial proportion of the IgM negative, IgD negative with B1, they proliferate. But their minimum proliferate for the adult populations. And also it's the statistic summarized. And it's also true for the air induction. As we showed before, you can clearly see that the air expression waned as the mice enter the adulthood. So this is the air expression level. So it's highly expressed in the neonatal thymus, but not in the adult. So, and also, so the activation means that the activation largely turn off in the adulthood. So that's also showing if you get IgM negative B1 cells, the expression of very activated phenotype. But when the mice, the 7 weeks mice, they do not express this activation marker. So, and we're also doing the BCR, single cell BCR sequence. So we know that we further showing that this air expression IgM negative, IgD negative B1 cells in the neonatal thymus are activated B1 are activated B cells, and they express very unique Ig repertoire. So we single cell sorting the air GFP CD5 positive IgM negative B cells from the day 8 to day 9 thymus. So we got from 4 mice. We have the 96 well plate, 4 plate. So in the recovery, we can recover 85 getting the single Ig heaven chain and light chain paired sequence from these cells. So you can see that different from the B1, the B2 from the outside of the thymus, they tend to express in the non-common VH1 families. So like the proximal VH family genes. It's very dramatically different from the regular B1 and B2 in the spleen and the peritoneal cavity. So they tend to using the less used VH families. So, this differ from the regular B1 and the B2 cells. So they express in diverse Ig repertoire and enriched with the uncommon VH1 family genes, which means long VH1 family gene. And it's also expected majority of the cells expressing germline encoded Ig. So there's a very minimum nucleotide insertion in the DJ and the VDJ junction. And also, very surprisingly, the majority of these cells have class switched. And they are switched to IgA and IgG2. So IgA is really amazing. Why they switch to IgA? Yeah. And it's also an interesting question to understand. And because although they have intensive class switch, there's very few somatic hypermutation. So taken together, so we are finding reveal a temporally restricted window in the earlier life for the thymic B1A antigen-presenting cell differentiation. So this unusual B1 activation and antigen-presenting cell differentiation depend on the CD4 T cells. And this T cell dependent B1 activation is selectively and transiently induced in the neonatal thymus. And the key question is that, is this T-dependent activation induced by the microbiota to which B1 exposed after birth, or by their initial encounter with the self-antigens? So currently we are testing this. We are testing these questions and try to analyze the germ-free neonatal thymus by collaborating with the Delaney, Dodi and Stanford University. So hopefully we can get the answer soon. So, but importantly, our also have the data showing that, support that this air expression B1 antigen protein cells in the neonatal thymus, they can induce a central B cell tolerance mechanism. About 20 years ago, Nicole Bangas in our lab have found a very interesting finding. They're showing that although mu-MT mice have no B cells, but somehow B cell cannot reconstitute in the adult mu-MT mice. So this is because the CD4 T cells generated in the mu-MT mice, they inhibit the reconstitution of B1 and B2 in the adult mu-MT host. And these inhibitory CD4 T cells develop in earlier life. And importantly, she also showing that transfer the B1 into the neonatal mu-MT mice, they can condition CD4 T cells to enable the B cell reconstitution. We further showing that we can repeat their finding, but further we're showing that the CD4 T cells in the mu-MT mice, the function, the inhibitory function is direct mechanism and also very potent. So you can see here, we transfer the B1 together with B2 into rag knockout mice. So rag have no T cells, B cells. You can clearly see both T cell and the B cells, no matter it's bone marrow derived B cells or the B1 cells, they are well reconstituted in the rag adult mice. But if you co-transfer this bone marrow progenitor or B1 together with the CD4 T cells isolated from the mu-MT mice, the B cells cannot reconstitute in the mu-MT. And importantly, also very few bone marrow derived T cells also cannot reconstitute in this recipient. Although they transfer the CD4 T cells can reside in the RAG1 host. So they indicate the CD4 inhibitory function is direct. It's not through other indirect mechanism. And also they're very important. Somehow they created some environment that can prohibit the T cell and B cell reconstitution. And we further showing that after transfer B cells into the mu-MT mice, the CD4 T cells in the mu-MT mice can respond to the transferred B cells. They differentiate into the IL-17 secreting CD4 effector T cells. So indicate they do have the T cells, CD4 T cells, can autoreact to the B cells and to differentiate to the inflammatory Th17 cells. So this is also ongoing study. We have now we generate the Th17 reporter mice under the mu-MT background. We really want to track the Th17 expression CD4 T cells after we transfer B cells to see what's the identity of these cells. So that indicate CD4 T cells in the mu-MT mice are autoreactive to the B cells after they encounter the B cells. So the immune tolerance means the immune system have to rapidly react to the dangerous agent, but need to ignore their own cells and also other mild molecule like in the food. So the development of the regulatory T cells, which can suppress this pathogen causing inflammatory T cells is underlying a wide range of the tolerance induction. So in the recent years, there's a very exciting study showing that this tolerance, including to the food and also to the successful pregnancy, is mediated by the Treg cells. And also the tolerance, they can also tolerance to the harmless gut microbiota. So the recent paper also showing that there's they call them the tolerance induction antigen printing cells in the gut. This is called the gamma T positive tolerogenic antigen printing cells in the gut. This is largely derived from the innate-like cells. So our preliminary data showing that the air expression B1 in the thymus antigen printing cells, IgM negative, also expressing this transcription factor. So it's very interesting to see if this transcription factor is really the marker for the tolerogenic antigen printing cells. Yeah, so still we are working on that directions. So very exciting finding is that we find in the Treg from the normal neonate, they can suppress the inhibitory CD4 T cells. Now they can enable the reconstitution of the B cells. So as I showed before, if you transfer bone marrow, B2 derived bone marrow on the B1 cells into the day 1 MUMT mice, the B cell can well reconstitute. So both B1 and B2 can generate in this recipient. But if you transfer the same cells into an adult MUMT, the B cell cannot reconstitute. This is because the adult mice have the B cell reactive CD4 T cells, which suppress their reconstitution. But if we co-transfer this B cell progenitor or B1 together with the sorted Treg cells from the normal neonatal, now this really magical thing happens. You can get in the B cell reconstitution. This is likely due to these Treg cells can suppress these B cell responsive CD4 T cells. So we can get both B1 and B2 from the recipient. So here just showing that both Treg from neonatal mice or from the adult mice, they can allow the B cell constitution in the spleen of the MUMT mice. However, if you co-transfer Treg from the MUMT mice, they cannot do that. So that's really indicate the neonatal Treg generation is abnormal in the absence of the B cells. And so indeed, we find the B cell deficient neonate have decreased the Treg generation, both in the, here just showing the thymus. It's the same thing for the spleen. And our hypothesis is that the air expression B1 antigen presenting cells can select these Treg cells, which allow the B cell reconstitution. So to study the air function, we also generate the B cell conditional knockout mice, so in which the air only deleted in the B cells, maintain normal in the other antigen presenting cells. So as you can see that we transfer, so yeah, let's just go through if you transfer Treg from the neonatal mice, they can allow the B cell reconstitution. From the adult, it's the same thing because the neonatal Treg can survive in adult animals, but they do not. Treg from the MUMT neonatal mice do not have this activity. But if you transfer Treg from the air control mice, like the Y-type mice, they have this activity. But if you transfer the Treg from the air B cell conditional knockout mice, they lost this capability. So that really indicates generation of the B cell tolerant Treg require air expression in the B1 cells. So putting together, unpublished study really identify the fundamental function of the B1 as the tolerogenic antigen printing cells in the neonatal thymus to induce the immune tolerance. This is a central B cell tolerance mechanism operate in the neonatal thymus. This is induced by the activated B1A generated air expression antigen printing cells, but it is mediated by the Treg selected by these antigen printing cells. So the model, our data supporting, and also we are still working on that, is that we propose that by interacting with the developing CD4 T cells, the B1 in the neonatal thymus, they proliferate, induce air, and internalize surface Ig and differentiate to the antigen printing cells. And these antigen printing cells then select the neonatal Treg that suppress the B cell reactive CD4 T cells, thereby conferring the tolerance to all B cells, and hence enabling the long-term establishment of the B cells. So the ongoing and the future studies that it's really amazing mechanism. So we still want to identify this air-dependent B1A and the CD4 T cells antigen-specific B1 cell or B cell tolerance mechanism in the neonatal thymus. And also, it's also important to realize that this mechanism. What is a mechanism that can promote their differentiation to the antigen printing cells in the neonatal thymus, but somehow inhibit such differentiation in the adult thymus and the spleen? And another very interesting question we really want to answer is that what's the role of the B1 play as the antigen printing cells that induce Treg to enable the establishment of the microbiota in the neonatal gut? Because we know B1 cells contribute a lot of the IgA, but it's still less studied field. We really want to go to that direction. And I will, acknowledgement, this study definitely is not impossible without the years support from the Herzenberg lab. And we also collaborate a lot with the other lab, including the microcytor lab, help with the RNA-seq, and we also got the CTO4 flux mice from the Shimon Sakaguchi, and also our project, the Treg, also got their collaboration. And the iRaportor is our long-term collaborator, so we did, it's very productive. And we got an air flux mice from the Mark Addison lab from the UCSF. And this is the pioneer person studying the air. And we got a carbohydrate microarray working with the DeLong. And we also do the collaboration with the Fudan University in China. And this study is supported by our 2 RON grant. And recently we got another one. So critically, it's come at a very critical time, so we can continue with that. Hopefully, we still can work. I'm still working with Lee, so we have another grant going on. So hopefully, we can still continue this very interesting study. I'm really enjoying this research. And I'm so apologize, I didn't prepare very nicely with the slides, but just showing briefly, this is my first joining the lab, and this picture attending the genetic retreat. This picture is taken by Lee. And also it's in the first Christmas party in the lab. And so it's really the family. It's amazing, awesome friends, colleague, And also I have 2 daughters, boy in this lab and a girl in this lab. So, and also girl with the Lee. We play the piano and to see the ballet performance, San Francisco ballet in San Francisco. And also 3 of us also dancing. So we also invite Lee to see our performance. So it's really the memorable, lifelong memorable memory. Thank you, everybody. Thanks for your attention. Questions?
Thanks, that was great. For the MD negative, have you done intracellular staining to find immunoglobulin of any type, and what are the isotypes?
Yeah, so as I didn't mention that, because in the CTO4 we find the IgM negative, IgD negative, CD5 positive cells. At that time we did the intracellular, we detect the IgM intracellular in there. So that's why we think it's most likely it's the internalized IgM. But for the thymus, we also detect intracellular IgM. But importantly, although we can detect the class switched transcript, we cannot detect the switched protein on the surface. Unlike the...
But what about inside? Can you see A inside?
I only detect the intracellular IgM inside. I didn't detect the other isotype inside. But the surface, they do not expression, although they clearly express in the transcript, the IgA, or because it's a single cell, so it's very clear.
No, right, yes. But message doesn't get you to functional protein. That's why I'm asking the staining question.
Yeah. But they didn't express in the surface until the mice get adulthood. But adulthood definitely can see the IgA and IgG2 on the surface. Yeah.
Any other questions for? Lee, would you like to?
I just want to make one little comment. This is all Yang's work. I come into the lab in the afternoon, she shows me this gorgeous experiments, and we talk a little bit about this and a little bit about that, and then I disappear for a week, and I come back, and she's got all this beautiful work again.
No, I think one thing I need to mention that I really enjoy writing with Lee, because that really sharpens your brain. Because every time he shouted at me, if I couldn't understand, nobody can understand you. So every time making me very clear in my
Yeah, I love working with you as well. We have a really good symbiotic relationship, but this is all Yang's work.
No.
It's beautiful.
I actually want to second that as well. Lee, you're a great mentor in terms of writing and preparing publications. I think all of us know that. I remember so many times sitting next to you at the computer, and that has really been a lifelong lesson for me as well. Thank you.
Yeah.
Yeah, sometimes it goes on forever into the night, but it's been very helpful. If there are no more questions, we have a break. I suggest we take a relatively short break since it's getting kind of late. Wait a minute.
No, this is not a question of Yang. Did anybody else park in the long-term lot? I'm getting like it timing out. And are they serious about towing cars around here?
Say again?
After 4, you're okay?
That's what I thought. That's what I thought.
Never mind, forget about it.
Okay.
Oh my God, I got something like 4:15. Okay, that's why it cut off at 4 o'clock.
So should we take a short break and maybe about 10 after reconvene? Okay, thank you.
start the last session in a couple minutes. It's been a long day, but we want to give some good, create a good audience for our last few speakers here. So, hi everyone. I'm Rachael Gerstein, for those of you who I haven't met yet. I'm at UMass Chan Medical School. I've been there for 25 years, and I was a postdoc with Len and Lee. I must have started in about 1994, and Kathy Seidel was a graduate student, and Aaron Cantor was a postdoc, and I think Steve came a little bit after that. And I came to Stanford to work with Michael Lieber, and my training to that point was using primarily molecular biology tools, which, I continue to have an interest in. And what I got excited about working with Aaron at first as a collaboration, and then I joined the lab, was really the power of marrying molecular readouts and cell surface readouts. So some people think of FACS as primarily a tool for interrogating surface protein expression, which is incredibly important. But the power of knowing the molecular processes inside of a cell using GFP or other reporters and using cell surface is what really got me excited. And so it was sort of a historical fortuitous situation. I joined the lab, and then we started working with GFP. Yeah. Okay. I'm just filling time really while people are filling their seats. So I'm the stand-up act. When GFP was first isolated and used as a tool in microscopy, microscopy has the power of the time integration of your eye looking at the subject, or the photography integration. It wasn't super bright, and so it had to be tinkered with in order to make it usable for flow cytometry. And so that was happening when I joined the lab, and Mike Anderson had the insight of how to make two different colors of GFP. And so that was really exciting to be able to have, that as an option, have two different readouts. And this is a bit of, kind of FACS history trivia, that we use the violet laser to excite one of the forms of GFP called VEX, for violet excited. And that laser at the time was, fairly expensive. And so Len said, you can't buy that laser just for this one thing. Okay, we have to find another use. So we scoured through it. Used to have to have catalogs. It wasn't online. Catalogs, for the different fluorescent compounds, and we found a couple. And so we found some dyes that would work with violet excitation. And, most commercial flow cytometers now have violet lasers to use violet excited dyes. We have the added benefit of having a violet excited GFP and a blue laser excited GFP. So just wanted to relate, so how do my interest grew. I created a reporter system for VDJ recombination, made a lot of different GFP transgenic mice for different purposes, and just really wanted to thank Lee for inviting me to join the lab. Kind of saved my career at the time, because Mike Lieber left kind of suddenly to go to another institution. It was a good situation for him, but in academia, I usually plan these things a little farther ahead. And so I had a month of thinking my career was over. And so thankfully it wasn't, because I was able to join the lab. And I was just so impressed with the diversity of interests and the diversity of people and the value placed on people and treating each other as colleagues. A little friendly argumentation every once in a while. But really it was just a model of how to treat people and how to care about people. And we all talk about the family atmosphere. And it's a testament, to that we're all here, umpteen many years later to celebrate our shared, our shared bond and our shared history. So I'm chairing the session, and with that, I'm going to invite Steve DeRosa to give a talk about his wonderful work.
Thanks, Rachel. So, well, here we go. Thank you. So, yes, I'm going to be talking today, giving a little bit of a different type of talk. Instead of talking specifically about my research, I decided I thought it would be useful for this audience to give more of a perspective of HIV vaccine development. Every time often I should say, when I tell people I work for an HIV vaccine testing network, one of the first questions people ask me is, how's it going? Or is there hope? Is there a chance we will have a vaccine? So this presentation is really meant to give you a sense of where we are now and how we got here and what has gone in the past. And talking about the diversity in the Herzenberg lab, I don't know if many of you know this, but when I joined the Herzenberg lab, it was a very difficult time in my life when I had decided to leave clinical medicine. And I decided to try research, and atypically, as a medical student at Stanford, I had not taken the opportunity to do research while I was a medical student, whereas a lot of people had done that, so I did not have much of exposure to it, that I decided I wanted to try research, and I specifically wanted to do research in HIV. And I found that Len and Lee were doing HIV research, and they were very kind to take me into their lab with virtually no past research experience. And obviously, my career has been devoted to HIV research ever since then. So I'm very pleased and very happy that you guys, it's worked great. It's worked out great for us. So let me, I'm actually going to be giving a slide set that was actually prepared by a colleague of mine, Jim Kublin. He's the executive director of the HIV Vaccine Trials Network. And I am working with Julie Malkorath in the laboratory of the HVTN. And I've been there for now nearly 20 years. And in January, it will be actually 20 years. So. Over that time period, we've seen quite a bit change in the landscape of HIV vaccines. So starting off to give you a bit of background about our organization, so the mission of the HVTN is to fully characterize the safety, immunogenicity, and efficacy of HIV vaccine candidates, with the goal, of course, of developing a safe, effective vaccine as rapidly as possible. Of course, my aspect of this is really to look at the immunogenicity, but today I'll be talking to you about the overall effort of our network and the not just our network, but the HIV vaccines that have really gone into further scale testing and many of the vaccines that were not effective and where we are going based on those disappointing results, unfortunately. So an overview of what is included in the presentation is I'll start by the history of the earlier parts of the vaccine effort. There are sort of two contrasting events that help us think about that. One trial that not only did not show efficacy, but actually showed just the opposite. It showed increased HIV infections in some vaccine recipients. And then around the same timing, a little bit later than that, there was actually one trial, and so far this is the only trial that has shown any clinical efficacy. It's referred to as the RV144 Thai trial because it was conducted in Thailand. The percent efficacy is relatively low, 30%, relatively low, very low actually when you think about it. Many of us have been hearing about these very successful COVID vaccines that have been developed so quickly that have had, effectiveness of 95% perhaps. So 30% is very low, but we'll talk a bit about that and how we interpret that. Based on those results, our network, in collaboration with other networks and other funding agencies, decided we needed to follow up and prove whether that result was actually valid. And in fact, we decided to focus on Southern Africa, where of course the epidemic has been and continues to be a huge problem. There, of course, we have clade C instead of clade B, so the vaccine had to be modified for that. And I'll show you the results of those studies, which again, I'm sorry, did not show efficacy. Two other large studies that I'll mention briefly also finished just recently, just earlier this year actually. One of them, these were vaccines developed by Johnson & Johnson, testing a mosaic vaccine concept and also no efficacy. So we're really at the point now where kind of back to the drawing board, and that's where I'll finish my presentation today. Really back to kind of what we expect. Previous vaccines have worked through largely the induction of neutralizing antibodies, and we weren't sure if we'd ever be able to get to that in HIV, but now we really realize that that's likely the goal we have to aim for. So why an HIV vaccine? People also ask me this, partly because now we have very good drugs that work well at preventing HIV, PrEP. And that works very well in developed countries, but we have to realize that, globally, that's not going to be the solution. And I think that is really we need to be thinking that way, that as a global solution, really vaccine is the only feasible solution. This is something actually, I remember this as well, and Rachael remembers this because I remember we went to some meetings together for ACT UP, and the reason Jim included this is because within the HVTN, we really pride ourselves on community involvement. The community is a critical part of our vaccine development, and we include them at all steps in the process, starting at the very beginning with concepts for vaccines. And this is just an example of, in the early stages of the epidemic, there was a concern about there were activists in the community concerned that scientists really weren't acting quickly enough and in the best interests. And this is an example of someone who's been in the news recently, Mayor Rudy Giuliani, when he was mayor of New York, and about his efforts to really fight the pandemic at that time. So one of the reasons HIV is so challenging is because of its diversity. It really is mutating so quickly that the vaccine can't keep up with the constantly changing virus, and in particular the envelope. And this is an example of these very nice sophisticated crystallographic studies these days that really nicely look at different envelopes from different strains, and the envelope is typically expressed as a trimer, and that's really one of the approaches we're taking now in HIV vaccine development, is to make sure our vaccine immunogens are actually presenting the envelope in the format that it's presented on the virus as a trimer, and that's relatively new. The diagram on the left is just showing the huge diversity. It's the standard sequencing type diagram that shows all of those branches, and the big changes over time. So it's constantly evolving, and you can imagine how challenging it is to keep up with that. And I'll refer to that again when we talk about some of the recent studies we've been doing injecting monoclonal antibodies as a potential means of preventing HIV infection. This is also another detail about another challenge with HIV is that we really do think that targeting the envelope, the so-called spike, is very important. We all are thinking about spike nowadays because we know the coronavirus is actually called the coronavirus because it's studded with these spikes on its surface. And interestingly, for HIV, one of the challenging aspects is that HIV does not have a lot of envelope on its surface. So that makes a challenge where you can't really cross-link those as easily because they're very sparse on the surface. So just another challenge that we need to face. And some vaccine strategies also to be thinking about, which is something that perhaps we've also thought about recently with COVID. We can think about, for instance, contrasting what do we want a vaccine to do. Ultimately, preventing infection would be great. But that may be too high a bar. So if we can get a vaccine that prevents disease, that would work just as well. I mean, you can become chronically infected with HIV. In fact, we know there are long-term survivors of HIV who are on treatment, who are very healthy. So if we can develop a vaccine that allows you to control the virus without treatment, that would be excellent. And then the middle category there is preventing systemic infection. And one of the reasons we've been thinking about that, there's a very exciting new concept that's been developed in rhesus macaques at Oregon, in fact, nearby where I am, by Louis Picker, using the CMV virus as a vector. And what it's showing is it's showing that it's actually may or may not be preventing infection entirely, but it's truly controlling infection, but you still see blips of virus. So it's really kind of preventing systemic infection. Exactly, we're probably seeing localized infection, but it doesn't become systemic. So those are three things we can look at. And again, I like to refer back to COVID, because it's an area that our lab has focused tremendously on the last few years, is that, you know, when we first designed the COVID vaccines, we actually used the clinical endpoint of preventing disease and not preventing infection. And looking back, I can realize why that was such a smart thing to do, because we know now that even though when we did not have the diversity in these newer variants, that the vaccines were preventing infections quite well, we know now that with the Omicron and other variants, the vaccines are not as effective as at preventing infection. They have some efficacy, but their tremendous efficacy is preventing disease. And we know that works well. So if you can prevent people from getting sick and dying, severely sick, that's extremely successful. So that's something to keep in mind. What do we want as our clinical endpoint? So moving on to the sort of perspective here of vaccines. In the early period from 1984 to 2000, there was some optimism that our traditional approach to vaccines, making recombinant proteins, monomeric proteins, something like, for instance, hepatitis B, and injecting that would work. And that the AIDS vax is one of the vax, that candidate, and those were not effective. So that was too much optimism. But we moved on from there, and then after 2000 to 2010, we were starting to think, well, if we're not going to have much luck making antibody-based vaccines, since this is a virus that's intracellular, the field, I should say, not me, I don't develop vaccines, I will only test the vaccines, but T-cell-based vaccines that were primarily targeting induction of T-cells, were focused on. And there I'll highlight the STEP and the RV144 trials to contrast the results from those. And this new era we're in, starting in about 2010 or maybe a little bit later, really is focused now on broadly neutralizing antibodies. And let me comment here, one of the reasons for that is it's actually quite surprising that it took this long into the epidemic for us to realize that people who are infected with HIV, a lot of people who are infected with HIV fairly commonly will develop antibodies that can broadly neutralize many different strains of HIV. So remember, those are the types of antibodies we would like to induce by vaccination. Now, in people who are infected with HIV, these antibodies are generally not helpful for them. They come on too long. It takes years. It really takes a long time. It's a developmental process. So unfortunately, by that stage, there's just too much diversity within their own body. But we are suspecting and hypothesize, and now have some clinical data to show that if these broadly neutralizing antibodies are there before infection, they could be very effective. So that's our goal right now, is to develop vaccines that can induce these broadly neutralizing antibodies. And in fact, they're using antibodies that have been isolated from people infected with HIV as the models for developing these. I mean, very, very specific models. We're actually going into the germline and trying to actually target the germlines that are producing these broadly neutralizing antibodies. And I'll show you about that. And that's where we are right now, is really the early phase testing of those vaccine candidates. Okay, then starting with these contrasting events. So the STEP, and we actually had a comparable study to STEP that was conducted in South Africa, the Fambili trials. We're using a vaccine developed by Merck, and again, primarily, it showed very excellent immunogenicity of T cells. But obviously, when we looked in a phase 3 testing, there was no efficacy in preventing infection, and in fact, there was higher infections in a subgroup of individuals. Turns out that that subgroup, this was primarily a study that was conducted in men who have sex with men, and those who were at risk of having higher infection turned out to be a subgroup of men who were uncircumcised and who had previous existing immunity to the adenovirus that was used as the viral vector, adenovirus serotype 5. So that was a very robust finding, that in fact, if you were naive, it did not have pre-existing immunity, and you were circumcised, there was no increased risk. There was no protective effect, but no increased risk. You have to remember that this is an important variable to determine, because all vaccine testing stopped immediately, obviously, because we could not be putting vaccines into people if there's any possibility of increased risk. So at least we have some understanding. Interestingly, even to this point, we really don't know the mechanisms for that increased risk. There's been a lot of work that's been done, and our lab has done some work, and we haven't really found the exact reason. But at least one idea is that the Ad5 vector was relatively more common. Many of us would have had common colds with that. So in terms of using viral vectors, we now would only use adenovectors that are much less common, so that most of us would not have pre-existing immunity. So that's one factor, at least, that we've also moved away from using adenoviruses to other types of viruses, although the Ad26 virus is one that is used in the Johnson & Johnson vaccine for COVID-19, and also their vaccine for HIV, which I'll briefly talk about. And then and another, at least, positive effect, or I should say it wasn't a positive effect, but at least it was reassuring that the increased acquisition risk was short-lived. So whatever increased risk the vaccine was inducing, at least it was gone over a period of a year. And after that period of a year, the increased risk in those participants was no longer a factor. It wasn't any higher in those individuals after the year. So this obviously brings up the issue of immune activation, and what's unique about HIV, of course, is HIV infects CD4 T cells, a part of our immune response, and in fact, it preferentially infects activated CD4 T cells. So what are we doing when we vaccinate someone? We're activating their immune system, and we want to induce CD4 T cells because CD4 T cells are critical for inducing antibodies. And so what we really are focusing now on is we really think that what we need is a vaccine that sort of balances that immune response, so that if we do induce a CD4 activated response, which we hope we will, it's balanced by a comparably large or higher antibody response, and perhaps also a CD8 T cell response. So that's one of the thoughts we have in terms of what went on with the STEP trial, perhaps is this imbalance where we activated the CD4 T cells too much, and we didn't have a balancing protective immune response. But like I said, we really don't know for sure what's happening. And keep in mind that, of course, HIV infection is happening at mucosal surfaces, and we can't always investigate what's exactly happening at the mucosal surface. Although in our network, we do collect mucosal sampling for some of our studies. So moving on to a positive result, the RV144 trial showed some efficacy, again, 31%, not huge, but it was clearly, and it was significant, and in fact, that 31% was based upon a longer-term follow-up, but if you actually looked in the first year after vaccination, the efficacy was much higher, almost 60%. So just to give a little bit of historical perspective, even at the time when these results were first released, there was a lot of controversy in the field about whether this was a robust enough response to follow up on, or could it have been a statistical fluke? Our own group participated in an immune correlates analysis that actually revealed an immune correlate. And what an immune correlates analysis is looking for is there an immune response induced by the vaccine that correlates with protection from infection? With those people who are protected versus those who are not protected. And in fact, we found a fairly robust antibody response for antibodies that are binding to a certain variable region of the envelope, the V1, V2 region. And again, we call it binding because we knew these were antibodies that were not neutralizing, but they bound to that region. So we suspected that there were non-neutralizing activities of the antibodies that were responsible for this effect, such as ADCC and other types of complement-mediated mechanisms of those antibodies. Interestingly, I won't get into this today, it's a little more complicated, but IgA antibodies binding to envelope had the opposite effect, that higher levels of those antibodies were associated with infection risk. And there's theories about why there's a balance between those two that I won't get into today. But some encouraging results from RV144, and again, in our own hands, looking at these correlates results, we felt even more confident that it wasn't a statistical fluke, that there was actually something induced by the vaccine. And I think I'm going to skip over this for now. And immune correlates are generally important, but I don't want to spend time since it's so late right now. So what our network, and in collaboration with other networks, followed up on the AMP study, on the RV144 study. And we ended up conducting a clinical trial in Southern Africa, HVTN 702, that was not efficacious. So unfortunately, we could not reproduce what was found in Thailand. There are a number of reasons. It could be, different geographic factors, different risk factors. Who knows? We don't know. But unfortunately, the results did not carry forward. Listed on this slide are also the two studies conducted by, with vaccines that were developed by Johnson & Johnson, and those are the HVTN 705 and 706 studies. Interestingly, the concept there is a nice concept, I think. One of them is called Mosaico because they're actually using a mosaic approach where we have collaborators at Los Alamos who have developed vaccine immunogens that in silico, they're able to take into account the diversity of viral strains and make immunogens that can hopefully induce responses that are going to be better at covering those various immune strains. So the concept did not work, unfortunately. And I'll be talking about the AMP studies in a moment. So let me just go forward, sorry, there we go. And again, I don't think I need to go into the details of this, but again, we followed up on RV144 and the results we did not find efficacy. And then 705 is one of those Janssen or Johnson & Johnson vaccines that was using the mosaic approach. And once again, that one did not show efficacy. And finally, 706, again, so a series of large vaccine studies conducted through the HVTN. But the other thing I do like to emphasize whenever I talk about our network in HIV testing is that I think these studies also prove the importance of testing in humans. There's only so far you can go with testing in animal models, and most often these studies are preceded by challenge studies in non-human primates. And those challenge studies over time have been improved to try to match the type of challenge you're going to see in humans. But once again, unfortunately, even though those primate studies showed that these vaccines are likely to have some efficacy, when we test it in humans, it doesn't go forward, it doesn't follow through. So I get emphasizing the importance for testing in humans. So 706, these results were just released, I believe, within the last year, very, very recently. So let's move on next to the next phase then. So antibodies, again, I'm talking to a crowd here of obviously who knows a lot and understands about antibodies, but antibodies used as passive immunization. So we're going to start there, and this is sort of a test of concept for whether or not neutralizing antibodies could potentially prevent infection. So again, there's things here which I probably don't need to spend time with this audience, but there's actually a relatively long history of using antibodies as passive immunization, passive transfer for protection. And again, so I won't, many of you know how these are used, but at the same time, I will just move ahead to studies that we conducted and this actually brings into context one of our colleagues, Mario Roederer, who's at the Vaccine Research Center at the NIH. In fact, I moved with him when that center was opened in about 2000, specifically Vaccine Research Center to develop vaccines, initially focusing on HIV. And they had actually discovered, as I mentioned, that there are these antibodies in HIV-infected people that they found that are broadly neutralizing. So they actually they isolated some of these broadly neutralizing antibodies, characterized them, and then made one of them into a clinical product. It's called VRC01, actually the first one that they isolated and started working up. And so ART Network, the HVTN, decided to test the concept, well, can we infuse people with this broadly neutralizing antibody, people who are at risk for HIV, and will that prevent infection, as a test of concept about whether or not a vaccine that induced those antibodies could do the job. And those studies were referred to as the AMP studies, and that's antibody-mediated prevention. And again, there were two of these studies. One was conducted in women in sub-Saharan Africa, and the other was conducted in men who have sex with men and transgender women in the Americas and Europe. And I'll skip ahead. These are the clinical sites to the results of that. And so overall, there was no efficacy. So there was no efficacy overall, except what we did discover is that we performed some quite sophisticated sequencing analysis of the strains of HIV that actually caused the infections. And looking carefully at those strains, we did find that there was protection for those individuals who were protected, who had viruses that were sensitive to VRC01. So the bottom line is that even though the VRC01 broadly neutralizing antibody wasn't broad enough, and you can imagine that if you're really trying to prevent infection in the real world where you're potentially exposed to any virus, you really have to be super broad. And that was the result of this. So we have a test of concept that, in fact, you can prevent infection with viruses that the antibody can neutralize. So that was the bottom line result. And what we realized from that is that one antibody on its own is not broad enough. So now we probably need to be using triple antibody combinations. And that's what we're currently testing in some of our phase 1 studies. And then, of course, we want to go forward and have vaccines that induce those triple antibodies. So at least we learned a lot from the AMP studies, at least. So just finishing up with our HIV vaccine portfolio in these early phase studies, as I mentioned, that we really are thinking that neutralizing antibodies are critical to induce. And I only briefly referred to the fact that these are very difficult to induce, extremely difficult to induce. So the work that's happening now in the lab, in the preclinical area, and even before that, is really, there are these very nice methods, nice concepts, I should say, for how can we model what is happening in an HIV-infected person that eventually leads to those antibodies. And they're now developing immunogens that can model that in humans. And we're testing these in what we're calling this discovery medicine program, so these smaller phase 1 studies that hopefully can provide us answers more quickly. And most recently, with the demonstration of the mRNA platform as being so successful, this has really accelerated our ability to test these concepts quickly, because now we can make RNA immunogens very quickly and turn around these trials very quickly. So it's exciting that we are now going forward with this phase of our program. And this is listing some of those approaches that I talked about. The one that's in green there is the germline targeting vaccine design, and that relates to what I was talking about, modeling what we see with these very specific antibodies that have been isolated from HIV-infected people, such as VRC01. Those antibodies use certain variable heavy and light regions in the human, so we're actually targeting immunogens that can actually target those regions so that we can make antibodies that use those specific regions. Interestingly, the other aspect of these broadly neutralizing antibodies in humans is that the very first version of those antibodies are not neutralizing. They need to be shepherded over time with mutations, and they get to become neutralizing over many months to years. So that's the approach we're taking as well for the germline targeting approach, is first target the germline and then do sequential immunizations to shepherd that antibody toward the mutations that we want to get to broadly neutralization. So you can see it's a sophisticated design strategy, and we're at the point where we're testing in humans now. We've only gotten to that first phase so far. So we've gotten to the germline targeting, and we've seen some success. So that's as far as we've gotten so far, but the other steps are in clinical testing. And then there are a few other approaches. The approach on the far right is using stable trimers. In the past, we haven't focused as much on matching the three-dimensional structure of the envelope to what's actually on the virus. So that's also a major approach that we're taking, is to really use these stabilized trimers, again with the hope of getting neutralizing antibodies. This is just an example of some of those broadly neutralizing antibodies that I talked about, and the fact that they target different parts of the envelope. That's the reason why we think using combinations of these antibodies will be important. So the immune system would be less, the virus would be less likely to escape if we attack it from multiple different angles. So that's where we're going. And we're moving towards this way. Like I said, haven't gotten there yet. This is a recent publication from our group, again, with that germline strategy and some sophisticated sequencing work done in our lab by my colleague Kristen Cohen, where we actually did document that we did get antibodies using the V heavy and V light regions that we wanted to, and with the right CDR lengths. So a demonstration of this concept, just recently published. And I'll finish with this other idea of new strategies as well. In addition to the strategies I just talked about, there's been some recent work, which I would guess probably follows up on older work, about vaccine strategies for inducing better responses. And here, because when you model HIV, any type of infection, your body is exposed to the infectious pathogen over time, not as a single bolus. So groups have now been looking at these fractionated dosing of the vaccine. So giving the vaccine on, every other day for 2 weeks. And in fact, that dramatically increases the antibody response. And in fact, most dramatically increases that response in the lymph nodes for your follicular helper cell response that helps the B cells. So it really is felt that this approach, again, how practical it may be in the real world may be something we need to think about, but certainly in phase 1 studies, we are doing it in humans. And we don't have those data yet available to show you, although I've seen the data and we are seeing quite a big difference. So some concepts that we're now trying. I think it's important that we're this is that study I actually just talked about, so I can't show you those results yet. And the last data slide here is just our HVTN pipeline of phase 1 studies. This is only those that are less than 50% enrolled. The list is actually even larger for those that are ongoing. So I think this is at least encouraging. Again, being in the HIV vaccine field can be very distressing because, and as you can see, the many vaccine trials that we've conducted that have not shown efficacy. But we are thinking towards the future, and I think we're fortunate to have a large pipeline of new concepts that are moving down the line. So I think I will finish there, just make a few concluding remarks here. And again, as I said before, I really do think that clinical testing is critical. There is a role for testing in animal models, but I think the two need to be considered together. As I mentioned, again, renewed focus on induction of broadly neutralizing antibodies that requires these novel, intelligently designed immunogens and iterative clinical testing. We need to be able to test these in the clinic quickly, and that's what we're aiming for. And in parallel, we are testing passive immunization combinations, and I should say that initially I pointed out that this was a test of concept, but we do have hopes that the passive immunization strategy can be used, at least in the developing world. Obviously, it's not going to be, in the developed world, I should say, it's not going to be perhaps very effective globally, that these can be used, and in the context of therapeutic use in HIV-infected people. And we are working with our collaborators at the ACTG to also test passive immunization as a therapy. And the protective role of T cells is not ignored. I should say that our network is also testing some vaccine strategies that really are primarily targeted towards cytotoxic T cells. There are just fewer of those concepts, but we are very happy to test those. Thanks to, of course, lots of different people. The HVTN is a very large organization. Of course, as I highlighted, Jim Kublin prepared most of these slides, and he's a very close collaborator of mine. And many of the vaccine developers that bring their vaccines to our network, we end up collaborating with them very closely. So they're not just giving us vaccines, they're collaborating with us on the design of the trial and the immunogenicity testing. Our funding, the HVTN, is funded by the NIH. We also get a fairly large amount of funding from the Gates Foundation. Those are our two primary funding sources. And of course, there's a large list of acknowledgments that I won't go through here, but of course, we always acknowledge the participants in our clinical trials. They're fabulous. Some of our trials ask for very unusual things. As I mentioned, sometimes vaccinations every 2 weeks, every 2 days, I should say, and sometimes mucosal sampling. Lately, we're getting lymph node aspirates, so they've been great. So I will end here. And sorry, I think I went over too hard. Thank you. Aaron has a question.
Can you comment on the that was great talk, Steve. Can you comment on the repertoire of the broadly neutralizing antibodies? You know, what V genes, sequences are they using, and are they shared from one individual to the next? You know, how common are they, and how did you pick the ones to use without that knowledge?
Those are great questions and completely out of my expertise. So we do know the answers to those, and I'm not the one to be able to answer it. Sorry, Aaron. But it's well known. These have been studied very, very nicely by a number of different groups. Michelle Newson-Swig.
But do you have a feel if there are a lot of shared components?
There are some shared components, okay. Yes. No, I think that's the reason why they're sort of hopeful that they can really target these specific VH, and these are VHs that are common. That are commonly used. So there is a lot of underlying basic research that's been done to justify, I should say, using this concept.
Okay. I'll go read a paper or two.
Yeah, no, there's a lot of papers out there, I know. Any other questions before we move on? Kathy?
Yeah, really interesting. And, I've talked to Mario over the years about this kind of work too. And it always strikes me that HIV's, of course, it's unique. It's, very mutagenic. It's very much like a tumor, right? It's going to change and develop resistance mechanisms. And so the strategy that it sounds like the field is taking now, where you're, doing one thing and then do another thing and do another thing. I mean, it's actually kind of like the therapy that works, right? You keep hitting it with something else and then it can't respond anymore. And so that's probably what's going to be required. And my question is, what kind of adjuvants are you using in these vaccines? Because it looked like it's really focused on antigens, but of course the adjuvants and getting that innate, Takeda, we have a big thing about innate immunity and how that leverages the adaptive immune system.
Yeah, no, the adjuvants are a huge aspect of this, and I should say that the director of our core operations, Larry Corey, has said in the past that our network is not focused on adjuvants, although within the laboratory, we have a specific interest in adjuvants, and one of the clinical trials that my mentor, Julie McElrath, conducted specifically compared 4 different adjuvants. And in fact, I'm also interested in that, and we collected samples at baseline day 1, day 3, day 7, so that we can specifically look at the innate immune response. So we are fitting in a little bit of innate studies within the network, but it isn't the primary focus of the network. And you're right to bring up the fact that adjuvants are a huge part of inducing the response. And the one that we found, by the way, is 3MO52 in our current testing. We haven't published it yet, but it's clearly dramatically higher, more activation of the immune system, the innate immune system. Whether or not that corresponds to a better adaptive response, we don't know yet.
Our next talk is by David Voehringer.
To quote Monty Python, now for something completely different. The science today, getting to witness it and watch it, it breaks my heart because it's so fascinating and so interesting and so intriguing. As I've left the lab, compared to what you guys are doing, I feel like a complete underachiever. However, there was something that Len told me. We've told anecdotes about how the Herzenbergs have made our lives better, and there's many facets that I can regale you at dinner tonight about how they've improved my life. But I came into the lab coming from a cancer biology background, and I thought I was going to cure cancer. I mean, granted, I understood it wasn't that simple, but that was my driving force. And I guess Len didn't see it in me. I don't know what it was, but Len pulled me aside one time and he said, David, how many shots of gold do you think you would have trying to cure cancer? You know how long the drug development pipeline schedule is? And this is back in the late 90s. And he says, it's like 10 years or so, more. So however many years you have left, that's maybe 2 shots at doing it. He said, you can take a lesson from me, and if you put your efforts into the technology side, the instrumentation side, you can have a much bigger and broader impact to the whole scientific community if you can develop technology that makes people's lives easier so they can answer questions more accurately, more quickly, and more on target. And I thought of that quote. I was at AACR this year. This is the first time this has ever happened to me because with COVID, we didn't go to meetings and everything. And so this was my first foray back into the real world. And I was walking up and down posters and everything whenever I could get a chance because I was the only person there for our company at the booth and all the stuff. But anyway, I left the booth every once in a while and walk up and down the posters. And I would see these figures of Western blots, and that's what I ended up going to do for the last 20 years is develop this technology, the automated Western blot. And to see the figures on those posters and see that technology there and realize that there's a whole new generation that is using this, relying on this, getting the results in 2 hours instead of a day or two, and it being analytical and things like that, it put a shiver down my spine, and it really, it was a cool feeling, and that came from the Herzenbergs. And so God, Kathy, you get emotional up here. It was really impactful. So I didn't know that Hiro was going to talk about this simple Western blot, or the simple fax machine, or this simple to use fax machine. I'm going to try to make it even simpler. And I risk falling on my face giving it to this audience because you're the world's fax experts and everything. And can you make something too simple? But as I said, I've been away from the community from when I left the lab. I ended up participating in the founding of a company and the technology, and then I shepherded it into the market and evangelized it, and left that a couple of years ago, and about a year ago. And for the past year, I've been traveling to all these different meetings. And the company I'm at now is a company that's preparing to launch a very low-cost, simple-to-use, kind of fundamental fax machine, a desktop microfluidics single laser cell sorter. And in talking, so what I've been doing over the last year, roughly, going to these meetings, is interviewing people across the spectrum of the type of people you meet in these meetings. Obviously, there's people that come by the booth that are fax people, and they ask all the fax questions and everything. But I've also tried to reach out to people that aren't fax people, but are really the fax customers, the people that come to use the cores and really have, these questions they're trying to answer, and what are they looking for in a tool and everything. And so I hope you bear with me as I kind of go through this. It's going to be very fundamental. Many of you are probably aware of some of these issues, but there was some unsettling aspects that I kind of came across, and I know that I'm going to be putting my efforts in the next couple of years to try to address some of these. And I'd like to get your feedback this evening to hear what you think of this. So I guess I control the slide. So the story, as I remember Len telling me, and Lee's confirmed this again, so there may be various versions of the story, and I'm getting older and my memory's not as good. But one of the things that drove the development of all this was the fact that Len just had bad eyesight. And so the famous picture, we've all been using these famous pictures. A couple of us have used some of these pictures multiple times. I think this is the first one I've seen of this one, but it represented really the foundation of fax, even though there's no fax machine there, or flow. It's Len looking through a tissue culture flask of the day. I mean, I sat there last night looking at that microscope and just, wow, that is old school. So this is the problem, right? His poor eyesight, maybe you could get better glasses or something, or just hire somebody to count the cells for you or something. Instead, what do the Herzenbergs do? They develop this to address the problem. And wow, what an amazing instrument. And addressing the initial question, but then more importantly, what it has enabled in science since then. And the irony about it all is that afterward, Len is still having to look through microscopes in a way. So he didn't even really solve his own problem. So I'm going to talk about fax and what we're seeing in the market and where we think there's a needed opportunity to address kind of the next generation of people coming into this community and trying to broaden the community out even more, because if single cells were relevant in the past, they're only more relevant nowadays. And part of the talk, and this is also going to be a transition into Wayne's session or discussion, is the software. And I imagine, and I'm not actually sure what the background of this picture is, but I imagine this is the early versions of what the software looked like, in that it was a printout from something, and this was FlowJo 0.0001 edition. It's a piece of paper from a strip chart or something. but yeah, mom and dad, scientific mom and dad. This is me. Oh, go ahead.
The first data retrieval were Polaroid pictures of the scope.
Okay, so let me show you this. So I go through this history of facts and everything, or very loosely the history. Everybody knows the history of facts here. But this was me like 2 days ago in a hotel room in Ashburn, Virginia, running a fax machine in my hotel room. I mean, I don't know if the staff there thought I was cooking meth or what I was doing with all these things I was bringing into the room. But it's become this easy. And Alan, to your point, if you actually notice, right, I can't see the pointer, but that little black thing on top of that, you know what that is? It's a jeweler's loop. So when you're in the field, you have to innovate and adapt and overcome. And so I'm doing sorting of cell mimetics, GFP cell mimetics and things like that to make sure the machine shipped and worked fine before I go over to Genelia Farms and run all these demos for them. And so after I do all the sorts, I'm sitting there with my little jeweler scope and a blue flashlight going from well to well to well to well to count, you know, what's the precision, what's the no cells, single cells, 2 cells. But yeah, it's got to be pretty simple when you can do a sort in 20 minutes in a hotel room. And this is, I think, where Hiro and I may be doing feats of strength tonight to figure out who's right or wrong. But I wanted an example of complicated fax sorters, and I actually put the Sony one in there. But you're saying it's simpler, and I'm not prejudging that. Again, I've been away from the field for 20 years. I've never used one, and maybe it is very simple to use. But I'm trying to make it even more simple. And getting back to that picture of Len at the microscope, making it more fundamental and asking the question of how do you provide a tool just to answer the question? And one of the things that I've come to realize and that we've seen, and the reason we've been on this development and gotten funding and backing and everything, is that, you know, again, single cell biology, single cell work is more important now than ever. But from that initial instrument, machines have only gotten more and more powerful, more and more perceptive, but technologically complicated, definitely more expensive. And in doing all that, you've kind of left this hole at the bottom of the market where there's really, what if you just want to get in and sort something simple? And so this is the only data I have in my presentation, and it's business kind of data, and it's actually not very accurate, it's very biased. But nevertheless, this is from these shows, this is a subsampling of something, and you don't have to pay attention to most of it. The point here is that when people would come by the booth, we kind of score them on what was their interest, what was their use, what would they want to do, those kinds of things. And obviously a big section, you know, if you have a fax machine at a show, cores come by and they got to check you out and everything. But that second quadrant there, the pie chart, the 18%, they're ranked in order and the color coding isn't relevant. But the single cell line cloning. So 18% of the conference people that we talked to at this point when this was generated, and it's held up as going to different conferences, and this is a broad spectrum of conferences, ASCO, or not ASCO, ASCB, AACR, some more flow-related meetings, a broad cross-section. You have a lot of people just wanting to do, the quintessential example was a young student that was doing CRISPR research. And he's got GFP co-expressed in there, and he wants to clone out these cells that he's just transfected, and he wants to make stable clones, and he just wants to sort them on a single color. And so that's kind of, in business, you talk about what's your beachhead, where do you want to focus on first and get the technology established there. That's kind of the beachhead we're going after. Because right now, you have these Ferraris out there in the world, these high-end machines, and people are doing GFP single color sorts in these high-end machines. And so that's like having a Ferrari and going to like the 7-Eleven to get a Slurpee and everything. You can do it, but it puts wear and tear on the vehicle and everything. And so we proudly embrace that we want to be like the Volkswagen of sorting. We're just the utilitarian sorter, inexpensive and simple to use. And And I think in doing that, it fills this gap of the next generation as they come up, so they're not intimidated by facts. Because let me now break to a personal anecdote and a confession I have never mentioned to anybody before, which is that I didn't know anything about facts when I came into the lab. To be honest, I still don't really know that much about facts, but definitely in this crowd I don't. This was when I was a college student, the junior, senior year at University of Texas. And here, I thought I was a Longhorn because I went to the University of Texas, but actually I was a Greenhorn. If anybody's familiar with the term Greenhorn, it's basically a naive noob that doesn't really know much. And that summer I got to intern at MD Anderson with a group that had just left Steve Rosenberg's lab, Liz Grimm and Jack Roth. And they had come down and were doing the first, Liz was the one who coined the term LAC, and Jack had done a lot of the IL-2 stuff with Steve on melanoma, and they were transferring it all down to MD Anderson for non-small cell lung cancer. And so my job as a college student was to go upstairs into the surgical suite, hold the petri dish, and Jack would drop the tumor in there, and then I'd go downstairs and break them all up and get the LAC cells. Point being, all this complicated stuff of isolating LAC and growing them with anti-CD3 and OKT3 and anti-TNF, and now I'm confusing myself, IL-2. None of that worried me. I could do all that stuff, cycle separations, I understood all that. I didn't even mind going in the surgical suite, I didn't faint. That was interesting. And I was injecting mice with tumors in the ipsilateral tail vein and growing lung mets, and then treating them with this immunotherapy. But they set me down in front of a early generation, the 1989 iteration FACT scan. That's when this was going on. And I just remember all they did was complain about, I'm an 18-year-old kid, and they're just like, this thing's always broken, it's costing us a fortune, da-da-da. And they're like, here, kid, you run the machine. And first off, it was amazing because this thing had something in it called a laser. And that was like, we didn't have CD players back then, we didn't have tape decks, and this laser was amazing that it had one in there, and there was a bunch of knobs and dials and how to use it and everything, and it scared me to death to run the machine, and it was very uncomfortable. So that was experience number one. I could still smile back then. When I was in the lab at that time, by the way, funny tangent even though Jean left, is that the woman who was a postdoc in the lab, who was whispering in my ear on how to run the fax machine, she was a postdoc, she eventually became Jean's first vice president of research at BioLegend, and now lives here in Redwood City, and we see her all the time. And she was the one that told me to go to graduate school instead of medical school. And she's like, you don't want to go to graduate school. The science will be much more interesting. So she convinced me to graduate school. So here I am a little older, so the beard's starting to grow finally. And this is still a greenhorn, though, second interaction with fax. And this was about halfway through MD Anderson. I was there for 8 years. And I was doing I was putting BCL2 in with GFP. So there's a BCL2 GFP construct that we had gotten from Clone Tech, I think it was, TET inducible. And we were going to clone them out with the GFP positive. So I went to the fax core, and now I'm a graduate student halfway through graduate school, and said, I need to learn how to use the fax machine. And they said, well, that's a $400 course, and it's a week-long class, and at the end, then you have to be mentored by somebody for a while, and eventually we might let you run the machine, or you could pay $50 and just give us your cells and we'll give you some clones. And so I went for option B, and sat next to the woman who ran it and was just in awe of what she was doing with knobs and tunings and all these things. I mean, imagine when I showed up at the Herzenberg lab when you had that 20-color MoFlo or whatever that was, and I was just like, oh my God, this thing's like launching the space shuttle. So my point in being kind of funny about this is, I really was intimidated by fax. And that ends up causing a couple problems, I think. I came into the Herzenberg lab because Lee came and gave a talk about HIV and redox, and I was interested in redox and cancer. and apoptosis. And so I joined the lab based on a science reason, not the facts reason per se, but obviously you come into the lab, you go to lab meetings, you're going to learn at least something about facts. Steve and Mario and Nicole, you guys were great teachers when I was there, and I've learned a lot from you guys. But if the technology is technically intimidating and not accessible, you tend to get people that just, like newer people coming in, aren't really going to be that interested in going in. It's not the path of least resistance. And then there are obviously people that are technically oriented and really want to learn about it, but I think you limit yourself. And so part of my theory here, or thesis would be, is that if this on-ramp was a simpler way that's less intimidating and more accessible in the sense of cost, like instead of a $500,000 instrument, it's a $50,000 instrument that you don't have to worry about breaking things on, can you teach some of the basic concepts of fax on an entry-level fax machine that does entry-level type things, which, back to that pie graph, is 17% of 18% of what people want to do anyway right now. I think the second danger that I've seen is that, or can think about, is that, and we all are interdependent on each other, but when you kind of hand things off to cores, you're handing your science off to somebody else, and you're not really sure what you're getting back all the time, and how competent they are in what they're doing. And then especially if you're not competent in that area, you don't really know. You just take it for what it is. So I think that that's the second important point, is that people, if they're exposed to fax early on and are fluent in at least the basic fundamentals of it, it's something that they can do on their own in many cases. And then lastly, human nature, people fear, or people, scientists tend to avoid what they fear. And the concept here is that it kind of limits what you will do with your science. So, I mean, I kind of avoided fax on purpose when I was still at MD Anderson because it was something I didn't understand. It was voodoo. And so I don't know if anybody will admit it, but there are certain techniques, or maybe we're all amazing scientists, but there are areas where you're like, I really don't understand how that works, and I'm just going to stay away from that. I don't know. This was at Gianellia Farms, and this is emblematic of kind of one of the problems I've been hearing about, is that we have a generation now of core facility folks that are all starting to retire. And this is Kathy Schaefer at Gianellia Farms, and she's kind of working with the new person that might be coming in to kind of replace her, or at least support the person that will replace her. And this was when I was there that day, and she's talking about drop delay, and they were talking about fanning effects and FMOs and all this thing. And then I'm over in the hood with this pulse photometer where you just drop the cells in the well and you push the thing in, and the cells sort, and you're done. And he came by afterwards, and like, he told me without her seeing that he was telling me this, he's like, oh, that other thing is just too complicated. This thing's really easy. And so I found a kindred person that had the same kind of experience I was having early on, was that, you want an easier way into the field. And so we're trying to address that. There's a number of companies that are in this space that is around that $100,000 price point, single laser, 3-color, just kind of a simpler fax machine. Namocell was acquired by Biotechne, who acquired the last company I was at, Protein Simple. NanoSelect has a really nice instrument with their Wolf sorters. And then we'll be launching our instrument. We just finished up our beta testing that Lee and Yang participated in and helped a lot with. And we'll be launching it, the first 10, in the back half of this year. But I think the benefit of having these simpler sorters, where again, it might not be very technically interesting to you guys, but a couple benefits. One, it offloads the burden on the high-end machines and in cores. The other thing I hear is like, we hate doing GFP sorts. Like, we have these high-end machines, and we'd rather be doing high-end work, but there's a lot of just people coming through, and if you're at big places like Berkeley and here and other places, these cores get filled up. And so if you can offload some of that more simple work to the end users and just have this fax like you would have a centrifuge or a microscope or a tissue culture incubator in your own lab, I've described it to the customer, as opposed to the user of fax, the customer of fax, that in a way it's just like a cell printer. It's just printing cells and wells for you. I think these simpler machines can be good on-ramp tools to learn on, and this will be borne out. I mean, NanoSelect and NanoCell can speak for themselves. I think that one of the things that we feel with kind of how we're doing it is that the gentleness and, trying to avoid Peter Lopez's, the whole sick phenomenon and things like that. He was out a couple weeks ago, and we were talking quite a bit about that. So we think that ultimately all these tools, ideally, you want the phenotype before you do something, then you do something to equal the phenotype after you've done that. And, with conventional FACS sorters, does it change the phenotype? There's people that are at some of these companies here that are very interested in this, and they've lived this, early HIV researchers. They've been in the AIDS community for a long time, and they're like, we know it rips the veils off dendritic cells, I think was one of the things that they said. Issues that are known about conventional FACS, and maybe some of these newer chip-based microfluidic sorters can help address some of that, but that's yet to be proven, at least in our case. And then one thing when I joined the company, it was a company of engineers out of Berkeley. And I don't know if it's late in the day, if I can say this way or not, but there's two things you deal with. You have the instrumentation, and then you have the software. And at the risk of being slightly inappropriate, the instrumentation, it looks great. It's like the opposite sex across a smoky room at a bar late at night. But the instrumentation ends up being the one-night stand. The day after you have it, nobody cares what the instrument looks like. The next thing is the software. The software is the marriage. And if you don't do the software right, nobody cares. I mean, the one-night stand's over, and then nobody will use your instrument and everything. And so that is something that and this is kind of leading into what Wayne, I think, will be talking about, maybe. We talked a little bit this morning. Is that on the left is, I forget whose version of software that is, but it looks complicated, and there's a lot of things on there to digest. Again, for everybody here in this audience, it's trivial, but for the uninitiated, that seems like a lot of stuff. We've tried to simplify it, ours in the bottom right corner, but you have to be able to live with the software. And so one of the things I've been thinking about recently is that, and this goes again to another Herzenberg-ism that we've heard today about, is always be asking the question, why are we doing it this way? Overturn the dogma. And the question I have is, we had it very complicated when I first saw it, and I was like, guys, this is way too complicated. We're trying to make this simple. So we made it graphically look simpler, but it's still got histograms and scatter plots and all this. And the computing power that we have nowadays versus even when I joined the lab, I think we were using DEC or VAX systems or mini computers. And even when you guys were doing FlowJo and with Mario and everything, we had the first IMAX. I mean, we have tremendous computing power nowadays. And unfortunately, I didn't get to go to the talk at CITO, but I know that there are groups out there that are asking the question, do we really need to go into all the nuts and bolts of all the different dimensionalities of the data to check every little thing? Or can we start leveraging the power of the hardware, the computing hardware, combined with the advances that have been made with machine learning and things like that to automate a lot more of this, where you basically have an Alexa version of a cytometer that you can come in and say, look, I want to sort GFP positive cells, and it starts asking you questions and you tell it what you want. And if it runs into issues, I mean, that was the question we were talking with Wayne this morning. I know this is pie in the sky, but if it runs into problems, there's solutions to those problems. And so how do we capture that type of thing? How do we solve those problems so that we can go back to that original slide at the beginning where Len wanted a tool that could allow him to perform a simple task that he was trying to do? Can we make a simple version of a fax where you don't have to be a flow jock to be able to run it, where you can just walk up to it and put your cells in and tell it what you need, and it does it in a meaningful, accurate, precise, affordable, informative way. So with that, I'll just touch on the last bit of this. And this is my colleague that joined the company. He was one of the founders of Essen and Incucyte and everything, and he took a sabbatical when the Sartorius bought them. Sohail and Yang, you met Sohail, and Lee met Sohail. Sohail's in the middle there in the blue, down low. He's always trying to stay out of the picture, but he took a sabbatical from science, kind of in a way, between Essen and then coming to work with us. And he was at USF and working at a community college, setting up a lab for kids that aren't going to go to a full 4-year college and everything, and teaching them basic fundamental lab skills, how to do tissue culture, how to do, microscopy, and one of the things is how to do FACS and cytometry. And, so these kids could potentially get jobs at Genentech or the companies around here as technicians. And so that's where I see, a lot of this going, because one last point I wanted to make was that of all those people that would come by the booth, it was interesting and it gave me hope in that about 1 in 10 of the people that were kind of fax people that were coming by would say, hey, I work at Genentech as an example, and we do lots of fax there and everything, but in my spare time, I teach fax at like my kids' high school. I go in there and give a course on it or something like that. I mean, I don't know if they have instruments there, but anyway, I just thought that was neat to see that it's incumbent on us of what has been bestowed on us to continue to passage this message down and try to broaden the field to bring more people in at that entry level and make people feel comfortable with fax. Because right now it's become really core-centric, and ideally to what Hiro said earlier is you want to democratize this and make it as simple as somebody using a microfuge or a centrifuge or a microscope. So with that, thank you, and I appreciate your attention.
Very nice talk. There is clearly a place in flow for a low-cost, easy, simple instrument. But I want to make a point. To me, there are 4 pillars for flow cytometry.
Right.
And we've talked about 2 of them.
Mm-hmm.
But the point needs to be made that all 4 of them were done by the Herzenberg lab. Right. They've been the leaders in all 4. So we've talked about instrumentation, we've talked about software, but flow cytometry would be nowhere without monoclonal antibodies.
Oh, that's true.
So monoclonal antibodies is what the 2 technologies had to come together. They luckily came together at the same time in the same place. The 4th technology was also spearheaded by Len Lee's lab with Vernon, which was the development of new fluorochromes.
Yeah.
So today, flow cytometry depends upon having the antibodies you want and having all the colors that you want. And that is also where simplicity, because we've tried to make simple machines, and I will tell you, a 3-color machine off of a single laser is not necessarily simple. Yeah. There's a lot of complexity in the data and the analysis and everything, but
it's taking it for granted.
It.
Like a CD player is not simple either. But if it wasn't for these guys and the vision that they had, and that's one of the things about developing technologies, is you never really know where it's going to go and where it's going to be taken. And it's really amazing how much they actually driven most of it too. With all the other patents and that are around the science aspect of it. I think that's the thing that's most unique. Is not only did they do, I'm only on the instrumentation side. I want to do the science side, but I can't do it. I'm not capable, whatever reason. Lee and Len did all of it. And for that, it was, I consider myself the Forrest Gump of science, getting to see these amazing people do amazing things, and it really has made my life exciting and interesting. So thanks.
Thank you. Just to respond to that and the issue, it is easier to do a study with 7 colors than it is to do a study with 2 colors. Right. And those people who think that it's easier, it gets more complicated when you get more colors, are those people who don't understand how to do analysis.
Yeah.
And the whole discussion is moot. I mean, it's not really, what you really need to be talking about is the non-specific staining, so that when you're looking at a small number of cells, you're having to really rule out a lot of other cells that don't belong in the population. If you're looking at something where you've got 50% of the cells, it isn't going to matter if you have a couple of...
Yeah, I guess my point is, though, but with a simple system, you can at least address the simple concepts that you build on, to your point of actually unleashing the power of the technology.
Look, you can do anything.
Yeah.
Okay? Is it worth your time to work with a simple... Suppose you have them side by side. Okay? Is it worth your time to focus on working with the two-color instrument as opposed to the seven-color instrument that you reduce to two colors? And the answer is it's not.
No, yeah, right.
It's much easier to do an experiment with a reasonable number of colors where you can rule out the cells that you don't want to rule in. The non-specific staining and other kinds of garbage that you have to rule out, that's why you use multicolor.
Sure.
And it's not, it really isn't a question. You use whatever you can get to rule out the garbage and see what you're interested in.
Mm-hmm.
And those people who try to do that in two colors and say, oh, I can do that, sure, you can, but not as well as you can do it in a seven color.
Yeah. Well, this is three colors with scatter, and it has the capability in the architecture to add a second laser and everything. The question becomes then, are we just copying everybody else and the price going up so that it becomes, because the other axis is the affordability of it. Are labs not buying these because they're over $100,000 or $200,000?
Well, that may be true, but you may be much better off to find somebody who lets you rent a little bit of time on their machine, like we do, basically.
Sure,
we run a facility. And there are facilities in most places. And the idea that you want to buy a cheap machine that you're going to use yourself rather than pay for the time on a facility machine is probably dumb as dumb as dumb. I mean, you really, if you're a commercial company, you can only afford to get a two-color machine. That's what you can afford to get. But if you get a machine that's three colors, you're going to find that you got better answers. And the answers are just better. But people don't know how to use multicolor, and they think, oh, I'm going to start with two-color, and it's going to be easier. And it's not. It's easier to start with at least 3 or 4.
Yeah.
So that you can rule out the garbage.
Well, ours is 3-color with scatter.
Yeah.
Take it.
Okay, my understanding is we have a short video that's gonna play before Wayne starts his thing.
Symposium for the lab. Hello, Lee, and everybody at the Symposium for the lab. Debbie and I are enjoying Butch Art Gardens in Victoria right now for our 40th anniversary. And we're hoping that the symposium is going well. And wish you all could be here and see the beautiful gardens and everything. Bye-bye.
Okay, Wayne is our next speaker. Looking forward to your remarks.
Okay, we're starting with this. We're actually doing 2 papers, but this is the paper that most of the work is being done by Connor and Stephen, who aren't here. And so I was just giving you the fact that it exists, and if you want to follow up on this, I can give you
Wait, ask if people can hear you in back.
Oh, I should have talked in the thing. Okay. This is, we're actually doing, the group of us, Connor and Stephen and me and Dave and Gunther and Lee, are actually doing 2 papers. And this one paper that's up there now is primarily the work of Connor and Stephen, who aren't here. So I'm not going to try to present it, but I can give you a slide deck that they sent and the links to the paper and so forth if you do want to follow up on that. And this is my talk. And this will to some extent follow Dave's. So it's not completely off the wall, but it's going to be a different talk, and it's going to be different in the fact that I'm actually going to say something about biology for probably the only time in my life. And so automatic phenotyping by exhaustive projection pursuit. Well, first of all, what does that mean in English, or what do I mean by that? So phenotyping, in what I'm talking about, the archetypical case that we're thinking about here is we're having a cell that's in some sort of genetic state where it's regulating the expression of molecules that we can label and then measure in facts. And then we assume that we have a sample that's made up of a mixture of phenotypes. And the fact that the cell is regulating it means that there's a most likely value of the measurement to be observed, and that as you go above that or below that, you're increasingly unlikely to find a cell that has that is that far away from that central point. And then each unique combination defines a phenotype. And then the problem, as I said, is the objective then is we've got a sample that's a mixture of these, and we want to figure out what phenotypes are present in the sample and enumerate the cells in there. And obviously this is a computer science problem, so we don't actually care if cells are involved, but just if the data follows this prescription of having a central, for each type of cell, there's a central value, and then it falls off on either side in all of the dimensions that we're using for phenotyping. And the way this problem is addressed is, if in geek speak, would be orthogonal projection pursuit. Projection, the idea of projection originated in the 16th century with Renaissance artists who developed the geometry of how to project three-dimensional objects onto a two-dimensional surface that was realistic. And then the mathematicians got hold of that and added more dimensions and other things besides perspective. And then by the 19th century, we had full-on projective geometry. It's orthogonal because in fluorescence anyway, you pick a number of dimensions in the data space, in the measurement space, you declare those to be orthogonal, and then you only use projections which preserve that orthogonality. And it's pursuit because it isn't a method that moves directly to the end in one step. It's a strategy, basically, of divide and conquer, where we find a way at each step of dividing a complex problem into two simpler problems, and then we keep doing that until we've resolved the issue. So currently, the way this is currently done in almost all cases requires a mixture of biological reasoning and spatial reasoning. Typically, people use their biological knowledge to pick a pair of dimensions, and then they plot the data onto that pair of dimensions, and then looking at the plot, you can see that one group is over here and one group is over there, and you draw a line in between to separate them. And if you've chosen a good boundary, separating the sample into two smaller halves will also separate the problem into two simpler problems. And then we go again and do that again on different dimensions until we've either found everything we were looking for or we don't find anything more to split. And you all know this. This is what this looks like. This is one of two experiments that we're going to discuss in the paper. I won't spend too long on that because you've seen that. And this is a more complicated one that we're going to use in the paper. These two are OMIP protocols. I don't even remember what OMIP stands for, but you all know. So currently, that's our best guide in higher dimensional space. High dimensional space is huge, and you can look around forever, and biology has been able to focus on things that get us through this high dimensional space. And in this process, in the process of drawing the boxes around, using the spatial reasoning to separate the sort of division you need to separate on a sorter, we identify these phenotypes spatially. But since you've used markers that you know, what you're also doing is you're locating the phenotypes that you find in your biology. This is a T cell, that's a B cell, that's a, whatever it is, eosinophils, whatever it has to be. In the process of doing the analysis, you are doing both a spatial analysis for sorting and a biological analysis, so you know what to do with the data. And another benefit that we discovered in doing this is that you ignore stuff. Those beads in the corner that are obvious, but everybody just ignores them, and the computer will find them. So that's another benefit of when or another aspect of when people do it as opposed to computers. And the drawback gives me an excuse to introduce my favorite Arthur Eddington quote. The imagination affects our experience of reality by limiting the alternatives to which it's compared. I think this is Eddington. I went to confirm this, and I cannot, but I remember this as being Arthur Eddington.
Say it again, Mike. Say it again slowly.
The imagination affects our experience of reality by limiting the alternatives to which we compare it. And in flow, again, if you're thinking of since you are using your biological knowledge to drive this analysis, well, you're a lot more likely to see what you go looking for. Okay, there's confirmation bias. And we have, 28 odd dimensions of which you've looked at a few, but you can't really look at every pair of every dimension. So we don't know what we're not seeing because nobody ever looked. And as you all really know, it's very labor intensive and requires substantial expertise to pull off. So, if the problem actually is a phenotyping problem of the kind I discussed, where you have these well-separated populations, then in fact it doesn't matter what order you take the gates. If all the gates are good, every case it has separated a problem into two simpler parts, eventually you will end up at the same place. And it does not matter if that's biology driven. If you have any pair of dimensions which separate things over here from things over there with nothing in between, that can't be wrong. So in principle, the order doesn't matter. And if you go down different pathways, you'll make different decisions on individual cells, but you'll always end, if the splits are good, you always end up with the same final phenotypes. So that observation that it doesn't matter what dimensions you choose, if it's good, it's good, led us to this concept of exhaustive projection pursuit, and which is basically try all of the dimensions and use the best. And obviously, you're not going to try all the dimensions, so what are we going to do? And so for every pair of dimensions, every possible pair of dimensions for the sample, we generate a plot, and basically we use a combination of techniques, modal clustering, density-based merging, and then there's a step I'm calling graph reduction. And I won't bother you with any of those, what they are. That's what the paper is about. But the point is we use those to mechanically generate a set of candidate splits on every pair of dimensions. And then it's obviously not every split is as good as every other split. And so we, and since this is a multi-step process, any mistakes we make up front will be propagated through every later step. So we want our good steps to be done first. And so we generate, a few hundred of these candidates for every sample, or at every step, in fact. And then we go through them and we score. The original idea was to score the separatrix on the basis of how many points or how many data points are close to it. Basically, if it's out in the weeds where there's no data around, nobody's going to argue about it. And if there aren't very many points nearby, there's really not much to have an argument about. And, you know, if there's more than you want, still, the one that goes through the least seems to be the best choice. And we kind of expected, and it turned out to be true, that that rule tends to split off a lot of small populations as it goes down the tree, and you end up with a very weedy-looking tree. So we added a balance factor that penalizes uneven splits, so that if it's 50-50, it doesn't have any penalty, and at 1 to 2, it gets a certain penalty. But when it gets like 4 or 5 to 1, then it begins to become increasingly penalized. And that does tend to produce gating trees that look a bit more like what people do. There's arguments on both sides, but everything I'm going to present is done with the balanced mode. And okay, so what's good about that? Well, we now have looked everywhere, or the computer has looked everywhere, so you know you're not missing something because you didn't look. And of course, the computer did all the work. You didn't have to do it. The bad is that the computer doesn't know the biology, so when you get to the end, it has no idea what those phenotypes are. It knows they're types in the sense that they're isolated from everything else, but there's no biological content in the analysis at that point. And that's the result of that. That's one example. This is, again, OMIP 47. I'm not going to belabor this now because I'm going to come back to it, but once you get a look at kind of what they have. Do I have a pointer? Okay. You can see that it doesn't really look like the gates that a person would draw. They go off in, like at the one in the middle of the top, it goes off to the upper right. That's because the method, when it doesn't have any data, it tries to draw a line that's equally far away from the populations that it has defined. But there's no data out there, so it doesn't really make any difference. That's all the art department. But if you look at the other gates, you can see it's choosing reasonable things that go between the populations. And here's a more complex one. I'll give you a minute to look at that. Okay, so what have we found out about this? Well, it often will pick very conventional reagents and do more or less what you would do, but it does not always. Sometimes it will find a different route, a quite novel route, to find the same final N populations that you will. This does not do everything. It doesn't do singlets versus doublets well. It doesn't do live dead. It won't do a dump channel. We haven't really tried it on cytokines, but it's likely that cytokines and stimulations markers are not always suitable either. It won't identify shoulder populations. It requires a distinct peak. But as I said, it provides none of the biological context. So if you're going to use this, there's going to have to be an additional step to provide that context. And there's a lot of details about this. This is primarily a biology audience, and I'm suppressing all of the computer science kind of talk. There's a lot of other things that I'm suppressing. And then, so the paper that we've written describing this does describe all of that. I mean, this is an informatics paper, so it does describe all of the methods of how we do this and why. But it's pointless if the biologists don't believe it. I mean, the informatics people have to believe it in order for it to, take off, but it's pointless unless the biologists believe it. And so there also is a biology component. And we didn't want the review to involve novel biology. So we picked 4 published peer-reviewed phenotyping experiments, and then we run, and these are For fluorescence cytometry, we did 3 of the OMIT protocols, which are well documented and publicly available. And for the mass cytometry, we're using a data set that we got from Genentech through Daria Orlova, who is also in the lab. And then we do EPP on the 4 experiments, and then we use QF matching, which was Daria's project. And then we can match the EPP phenotypes against the phenotypes that were in the reference experiment. And in this case, since again, EPP often goes further than people do. It can divide things into more populations than the people did. We identify 1 or more populations that are the best total match for each of the reference populations. And then we have to compare them. And so when are 2 phenotypes the same? This is not a statistical test. We're not trying to reproduce some specific test. We're really asking a biological question. We have 2 different analyses of the same data. When have they identified the same phenotype? And so if we have 2 sets that we think represent the same phenotype, that the matching has said these 2 things are the closest match, how can we estimate how close that match is? And what we've used is something called the Jacquard similarity coefficient, which is the number of cells that are in both, that are common to both analyses, divided by the number of cells that are in either analysis. So it's the and over the or. And if you actually plot that out and look at it, you can see that it's pretty easy to generate some pathological cases where you can get a pretty decent Jacquard score where you wouldn't believe it for a minute. And the basic difference is the outliers. When you're comparing 2 phenotypes, you don't care about the cells in the weeds. They could be dead or dying or whatever. They could be maturing into and out of the state. It's the cells that are in the main population close to this central valley that are the ones that really make you agree that these are the same phenotype. And in order to get at that, we came up with a central similarity thing where using Zipf's law that 20% of the cases cause 80% of the problems. We find the most concentrated 80% of the merged sample and then take the similarity coefficient of those data. And in the paper then, this is one example. This is yet another OMIP that we do 44, and this one is pretty complicated. And you can see the orange lines are the population from the reference gating, and the ones below it are the ones that QF matching has assigned to it. And you can see that most of the time in this experiment, we find several populations that all together make up one of the reference populations. And this is a mixture of things. Some of them seem quite, to my eyes, seem quite literally, well, some of them are things that are visible in the paper that, again, they just ignore, they don't talk about. Some of them are things that aren't visible in the paper, but that they look pretty decent. And then there's some small populations, and there's, I believe in this one, there's one that turns out to be a population of spurious beads, and there's one that's pretty clearly doublets. So there's no pre-filtering of the stuff that people do on that. So you do need to look at this with some care. And this is my biology statement, okay? I'm saying that this thing really does biology. The algorithm is using the same spatial reasoning that you do when you draw these plots. It's not identical, but it's basically, go between the mountains. It produces a conventional gating tree. It can be reviewed. It can be edited. It can be used to sort. So it's not any different than what you're using now, except that it doesn't have the biological annotations on it. And so I contend that you could start, instead of starting with the single cell data, you can start with the phenotype data. And in the process, of course, you have saved a lot of work. But we also need this additional annotation step. So if that's true, and the other thing, I mean, you know, the paper and the literature are important. We have to do that. But what we really want is truth. And in biology, rigor is review. So do these gating trees pass review? So that's my question to you, and I will put them up larger so you can look at them for a minute. And this one is a pretty tight, do I have a pointer? I don't think I do. If you can look at this, this is basically an isotyping experiment. It's IgG1, IgG3, IgA, IgD. And the top two are IG1, IG3. IgD is in the middle one, IgA is in the bottom left. The two in the lower right are both splitting on CD27, and the middle on the left is splitting on CD38 and CD27 to pull out the plasmablasts. And, you know, if a graduate student came to you with that, would you say it was wrong? And this one is a good deal more complicated, but again, if you were reviewing a paper that said that, would you say it was wrong? And if it can reliably produce a reviewable, if it can reliably produce an analysis which passes review, then it's doing biology. And we've separated the spatial component of that logic from the biology component of that logic. And this is fast. This takes a few seconds. It takes longer to read the file than it takes to do this. So if you accept that, then where do we go? One of the capsule definitions of informatics is that informatics is the answer to the question, what is the difference between data and information? And the short answer usually is structure. And EPP extracts a lot of the spatial structure from the data, from the individual cell data, but not all of it. Using something called the Kuhlbach-Liebler divergence, which again I won't otherwise explain, we can identify phenotypes that have more structure than we have accounted for. So if there is a phenotype thing where it couldn't find an additional split, but there's a big shoulder or there's a lot of outliers or there's some other additional structure in there, the KLD can probably tell us where we need to go and look deeper. Another thing we can do is, I mean, at the end of this, you basically have a big table of expressions and populations, but you know, nobody wants a giant Excel table. That's not really going to be useful. So in order to give some internal structure to the sample, we can use numerical taxonomy to discover relationships between the phenotypes. And a third thing we can do to extract more information is we can use something called Mahalanobis distance, and that allows us to identify things that are outliers. We can identify things that don't look like they fit any of the phenotypes and gate them out. And you know, the crud around the edge we can get out. So as I said, so that's what we can do further on the spatial. But EPP doesn't really give us any handle on the biology. So somehow or other we have to solve that problem. But starting from the phenotypes is an area where informatics as it exists has a lot more traction than it does on the single cell data. There's a lot more information there, and it's structured in a way that's a lot of existing techniques can be brought to bear. But it's, not what current software does, as you all know. So there's going to need, in order to actually take advantage of this, to reduce the amount of labor, there's going to need to be significant software development to make use of this. And I don't mean that I can specify what to do. I mean, there's going to be real development, research, real thinking about what's going on there. But you've moved, there are departments full of graduate students in informatics that would love a better defined problem than clustering to beat their heads against. And so I think there won't be any problem finding people that want to do that. The gating tree, we need it because we sort on it, we review on it, it's still got to be there, but the gating tree isn't giving you your biology. And the gating tree historically has been the center of analysis, and it needed to be. But so all the biology has basically been built on the framework of the gating tree. And somehow or other, we're going to have to do software development that is focused on the biology without the support of the gating tree. And I don't know what that's going to be, but it's going to be a very different sort of thing. And can I go back? Okay, so what do we do? Okay, so what does the brave new world look like? And so this is my first attempt to try to exploit some of these things. This is using the numerical taxonomy. The tree on the left is not a gating tree, it's a taxonomy tree, in which the things that are closely related are things that have closely related phenotypes. The phenotypes are similar. Currently here I'm using city block distance on logically scaled data. So basically this number is the total fold change in expression of any of the regulatory markers up or down. So it's a sort of a stand-in for the amount of regulatory work it would take to go between the two states. In the circles, the area of the circle is proportional to the size of the population, and they're sorted large to small within each clade, or they're not clades, they're taxa. And then the open circles are 100x magnified, so you can see the small populations. And then the circular spots are the marker expression, where warmer colors are more expression. They're labeled on the top. This is actually a live, this is not, this is just a picture, but the phenogram itself is live. You can get a legend so that you can read the reagents against the line that you want to look at, and you can resort the reagents so that they make some more sense and can visualize visualize different biological things. And this doesn't slavishly reproduce the biology, but in the cases where I do understand the biology does seem to be reflected in the trees. The B cells clump in one branch, or maybe there'll be two branches that are T cells or something like that. It's not, and then of course there's the doublets and the beads that get thrown in there too. So you have to look at it a bit. But it does seem to actually be reflecting something about the sample. And let's see, I've told you, yeah, you can sort it and so forth. So that's just sort of my, one concept of how this might look going forward once we've not driven everything in front of the gating tree. And that's it, unless you want to look at the samples again.
So, as I understand it, right now what you're showing us is the analysis of one sample.
Yes.
But what everyone wants to do is compare sets of samples.
Yes.
Healthy and diseased.
Yes.
So and you want to be able to look at all of that data in one shot, and then have a strategy that lets you look at the populations and describe differences. Can you do that? Or do you have the potential to do that?
I think there's the potential to do that. lining up these populations among samples is like one of those things. That's one of the things that informatics probably can do. And labeling it with what the different things are is something that we can do, but it's going to take work and it's going to take software.
But I think, the simplest thing is, take your simplest OMIP, okay, and look at a data set where it's been applied to two different populations. I mean, that must be out there for all these things that are, being published.
What?
You're already doing that.
And then take that and use your program and see, are you learning new things, and can you look at that data in mass and then come up with a gating strategy that you can apply to each of those samples. I mean, that's what we want to do, right? When we develop our gates, we're saying, what is this population and how does it differ in one mouse versus the next?
Right. But I'm not clear on what you're asking me. I mean, the thing is, it isn't necessarily going to come back with the same tree on another sample. It should end up with the same endpoints, but it's not necessarily going to make the same branches to get there.
Well, I guess it seems to me that you need to have a universal tree that you apply across samples.
No,
and in some places you'll have gaps, in some places you won't.
Why does it need to be a universal tree?
How do you do a comparison of what it means from one sample to the other?
Well, there is going to have to be an alignment process where you align I found this phenotype in sample A and this corresponds to this phenotype in sample B. That definitely will have to be done, but it doesn't have to be the gating tree. That's not the only way to solve that problem.
Well, you can be blind to the data tree, but it could be whatever that it doesn't, I guess, matter how you get there, but the populations you have at the end, are you able to compare them? And I don't see how this is a tool for that, and until you get that, I don't see how a biologist is going to want to use this.
That's what I'm saying, is this is an enabling technology, but it does not, it does part of the problem, but does not do all of the problem as it's currently stated. But if we did complete that loop, there'd be a lot less carpal tunnel syndrome in the world.
So I think to Aaron's point, and I know I've been frustrated as we've gone to UMAP and all these dimensionality reduction things with the clusters. And I remember the first lecture I heard on it says, oh, by the way, every time you run this program on this data set, you'll get a different answer of clusters. And as a biologist, that's really not good for me.
Yeah.
I need to see something reproducible. I need to see something reproducible so I can make comparisons. But I have a different question, and that is, everything you showed there and all the OMIPs and everything, those are basically binary markers. It's present here and it's not present here. And you're talking about the difficulty of working with shoulders. And I'll argue that most of biology works in the area of shoulders of that. It was easy. We discovered CD8, CD3, CD4 30 years ago. The differentiation of markers that, for example, there's certain markers, let's just take CD45RA. It goes over 1,000-fold. And it looks like a smear.
One of these has CD45RA in it. I'm sure we do that.
But in terms of understanding, I guess what I don't want to see is people to stop looking at. In other words, as you say, I don't want a list of all the different populations. I want to go. I'm a student of Lee. I want to go back and look at the raw data. I want to see the distribution. I want to see how it's clustering, because again, you're always going to have a cutoff point. It belongs in this group or it belongs in that group, and you do lose a sense. To me, you lose a sense of the valley, and that just may be that I'm a Luddite and I don't know how to look at things in these new ways. But it is a difficult transition for me as I look at data.
Well, it does not do all experiments. That's really clear. But if it is phenotyping in the sense that I defined it, it will do it.
And I'll make one more editorial, which is, and this is, software never discovers biology. Software discovers populations, which you then go back and do your experiments on to understand what is the biology of that population.
No,
and then you bring that back to your next analysis.
So these are mathematical phenotypes. Would you call them biological? Would you make a discrimination between a mathematical phenotype and a biological phenotype?
That's a good point.
these are the same as assuming there's a clean gate, these are exactly the same populations you're using now. Other than maybe the outlier cleanup, or again, you draw the thing so that you eliminate the outliers, and that might be another step. But there's no difference in the gate. I mean, it's a perfectly ordinary sortable gate, and the result is the same.
I'm on your side.
I mean, it's fully aware that it only carries it halfway to the biology. Okay, that's very much true.
But your point earlier about confirmation bias, and
yeah,
this takes that out.
Oh yeah. Yeah, and you don't have to worry if you've looked everywhere or if you haven't yet found something, because it has looked everywhere and it hasn't found a better alternative than what it's showing you. So you don't have to worry that you haven't found it.
Fit your narrative around the data as opposed to fitting the data around the narrative.
Say again?
There were some gates on the OMIP things that I did not agree with.
Okay.
And I'm just wondering, is there benefit to having a generative part to this where you sort of teach it, okay, in this kind of situation, it's better if you take it to the left a little bit. And of course it's subjective, and if you asked all of us, we'd probably draw different gates.
It is somewhat biased against small populations because it's demanding that there be enough data that there's statistical proof that there's something there, that it's not just looking at noise. Okay, so it is somewhat, where it draws its edges are probably not exactly where you would draw them. Especially where it's, where there's
spare.
A good dip, it'll do what you do.
Yeah, that's obvious,
where
but
it's
flat,
where
it's like exactly.
It will tend to be biased against the small guy. But it finds the phenotype. It's not necessarily that all the single cell assignments that it makes are the same ones you do, but it found the phenotypes. And it can characterize those phenotypes in terms of the mean and the covariance. So it can give you quantitative guidance as to where to set that. If you want to sort those, it does give you quantitative guidance as to where you could set a boundary to get the best sorting.
Yeah, it's probably a better gate for sort than for a percent, population.
It's not necessarily going to be the best gating for your purpose, but it will find the phenotypes. It should always find the phenotypes if there's enough data. We have, it depends on the size of the sample and the size of the subpopulation. And it doesn't take everything. Like I said, it does do statistical significance to make sure that you're not just looking at noise. But it should find the phenotypes. And it gives you enough information that if you then want to make a mixture model to see what the true percentages are, or you want to find the best separation, it gives you the covariance matrix, so you can find the point between them that's the best separation. And then there's also all the other things that you can do. You can, once you've got these populations, then you may want to look at the stimulation markers or calcium or whatever else it is that isn't phenotyping. And there's all kinds of other things that you can drag in, but in terms of giving an unknown sample, you don't have to go around and beat the weeds and find the phenotype. And yeah, there's some things will be shoulders, and the software eventually will have to cope with that in reality. But the point basically is that if, to the extent you can use it, you spend a lot less time mousing.
We should actually have a good look at it. We should all.
Can I bring in the back?
Yeah,
I don't think I can go
that
back
would be good.
anymore.
Yeah.
Okay.
Oh, good. Yeah, I was going to bring a couple other points there. Okay. Yeah, one of the things that
So that one?
No.
Which one did you not like?
It wasn't on this slide. It was on the other slide. So like the middle left one, I would have drawn it a little bit more to the left.
Yes, but it found the phenotypes.
It did find the phenotype, yeah, but I'm also interested in the frequency, right, potentially.
You mean panel B1 there.
Yes, B1.
Yeah. Yeah, I mean, this is making a statistical thing to find where there are the fewest the least ambiguity. That's the problem with this. If you've got a perfectly clean one, it doesn't matter. You can move it doesn't matter. If you've got something that's got, where there are events, there's no way to draw a line that avoids the nearby events. And the question is that then fundamentally you have ambiguous allocations.
Yes.
And if you see, even like that bottom one, the GA there, there are things that are, you're going to qualify there as technically IgA positive, but are going to be on the left side of that cutoff, and vice versa. And like on the lower right one, there's a lot of things there. You say, I wouldn't believe the stuff close to that individually. And so that's something that people just take into account as part of the ordinary experience of analyzing this stuff, is to realize that you don't have a pure population if you've had to draw through a bunch of stuff.
Right.
And you're not going to get a perfect sort or whatever in there. But anyway, but it is based on things that there's a statistically supportable
argument for why to draw it there.
Evidence that this is not one thing, and that you can have some definition of the best estimate of dividing them, and so that's a
and there isn't necessarily a best anscer. I mean, that's I mean, in any of these things that you do routinely, you don't always agree with what they did. You want the gate a little over here, they want it a little over there, or whatever. But if it survives review, it isn't serious enough to challenge the conclusions that you're reaching to. And I'm saying it survives review in that sense.
Yeah.
It's not ever going to be exactly what some person would do, but it's not incorrect.
Yeah. Well, the solution is to find a better marker.
Yes.
A clean separation.
Hang on, guys. That's exactly what this allows you to do.
Or perhaps a better instrument.
But David, this allows you to evaluate your markers and find which ones are better. And you can't expect them the first shot to do it. But it's what we do.
Yeah, sometimes it does find a better.
Yeah.
I mean, it's typically is stuff it just wasn't the way you did it, but it will be cleaner.
No, and some of the things we've looked at, it's clear that you could do a little bit better job of separating the things than they actually did, given their data. If you used a different choice of reagents where the separation between positives and negatives in this dimension is not as good, and then you might choose a different order of trying to separate it and do well. The other thing I think what Aaron was saying about the comparison kind of things, that's that one thing that Wayne showed that had the table with the orange lines across it there is actually a comparison of that sort. It's a reference in this, but it would be the same if there were just two samples and we say, okay, how do we align these samples? In this case, just because the EPP went further down in most cases, it's not equal, but in samples that were really more parallel, you'd expect to get pretty much one-to-one pairings. You'd find one thing in each and find them. And you wouldn't necessarily have to go the other route that people commonly do to try to make a UMAP make sense or whatever, of aggregating a number of samples, trying to get a common display, and then showing the individual sample data against that common display, which is a way of...
My thought would be, okay, here's your gating scheme, your proposed gating scheme, apply it to many samples.
Yeah.
But I know from sample to sample, just the noise, even on your bouncy mice, all at the same age, you're going to have some variance. And so, one, you can just pull those data sets together, from multiple animals and do your initial gate finding.
Yeah.
Or you do them separately. But at the end, I want to be able to compare those.
And then you get your software thing to go ahead and take the individual sample ones and try to move them, or optimize them. And there are people working on this stuff too. Yeah, that's a version of things that have been done.
Healthy mouse, diseased mouse, okay? Put them in one data set, make your tree, and then apply your tree, the same gates separately to each of those two mice.
Yeah. So,
See
and
if
this
you get the same populations or not.
Yeah, and this goes at it the other way, of going de novo with no preconceptions on each sample, and then lining them up afterwards. And a little bit like here, this is really doing a match of where are the same events, where do they fall, and so that's QF matches. Yeah, that's exactly what that was designed to do. Yeah. Yeah, and also that the Wayne's sort of quick phenogram that there at the end there too. That's the thing. He sort of presented this as a weakness that you don't have automatic allocation or annotation of stuff when you go in, but you're expressing your preconceptions in order to gate and know that, oh, I'm going to go find this population using these markers and stuff like that. And this, if you know which markers should be high and low on a given sample and like that effectively is their allocate. If you know what's going on, looking at it, you can tell which population is which of those things. And that's the thing you want not to do by hand anymore, but you're doing it from a much more compact thing than a million cell data set.
And where this becomes really important is in the clinical world where they do minimal residual disease. And you want to know, is this little population truly unique, different? How much is it? Is it similar to something that existed before, or is it unique?
I mean, there is a software that does that. I can't remember the name of it. It's from Salamanca, Spain, right? And they use Euroflow, non-parametric analysis.
You know that one, Wayne?
Oh, you do?
She just mentioned something that was put out by people in Salamanca, in Spain.
It's the Euroflow group.
The Euroflow group. There is, you say.
I can't remember the name of the company, but it's from Salamanca, Spain. It starts with an I, and they have 7 panels that they worked many years to develop. And then they have a bunch of normal, and then a bunch of disease. And they use non-parametric analysis, to do what Aaron was asking. And then you just do the flow, use the exact same panels all the time. And then through non-parametric analysis, it says, oh, this is CLL, this is ALL, this is normal. And it's looking at all these different populations, and they're trying to use that to supplement the pathology. It sounds like Alan said BD bought it, so I guess he's trying to remember the name also.
We're kind of coming out of a little more of a academic research origin where not doing large numbers of things in tight parallel.
I'll answer any
Not
questions.
like a lot of clinical is a different thing, but we historically we work mostly on research where you're not doing huge numbers of the same
things.
things,
Muffled by
and
not
you want to look at it and then make sense of the sequence. So the idea of analyzing each sample individually, getting and then getting the phenotype results or whatever. Also, that last bit, the divergence thing, maybe turn out interesting too. That's sort of thing that tells you how much residual shape is there in this allocated. Once you've gotten this population and as far down as you can say there are statistically justifiable separations, how much smear and how much non-normality is left up there, basically. Is it indications there's more here, but it doesn't have, it doesn't qualify as a place to find a division, but it's a place you can look to see whether you've got a shoulder there that you want to do, or whether you've got some junk that's out there away that you want to prune off.
Right. I'm just going to mention that the last thing on the program was Lee was going to give some remarks, but just given the hour, the executive decision is that she's happy to answer some questions. You know, if people, we've had a lot of different topics, a lot of different great, you know, memories and tie-ins to things that have gone over the years. Going to come and ask questions, come and ask them. Otherwise, you know, we're going to conclude the really formal part of the day and adjourn in a little while to dinner.
Here for tomorrow afternoon's meeting? Where are we meeting?
Lee's.
I think it's at
Lee's house.
More informal.
Do we have any more things we want to discuss? I mean, it's simple, informal, and anything anybody wants to bring up, they're welcome to bring up. If not, we'll go have supper.
How are we doing? We need to be out of here at 6, right? That's 6:36, so we need to be out.
They said 6:30, which
Yeah.
we're past, so we need to
Yeah.
let the guys,
That's time.
let the guys pack up.
Everybody was fortunate.